Institute of Science, Bangalore. Welcome you, sir. And the second speaker is Professor Amitabh Jha, a renowned scientist from Department of Chemistry, Acadia University, Canada. I welcome you, sir. It is a pride for us to have two chairpersons with us, Dr. Aditya Saxena, Associate Professor of Physics and IQAC Coordinator, Deshbandhu College, and Dr. Aparna Shekhar, Assistant Professor in Department of Chemistry, Deshbandhu College. All the participants are requested to remain joined till the end of the session. Feedback form is mandatory to fill for each participant, which will be shared in the chat box towards the end of the session. May I now invite our chairperson, Dr. Aditya Saxena, to conduct the session further. Over to you, sir.
Aditya sir,
please
unmute yourself.
Thank you so much, Dr. Parul. And it is indeed a pleasure and an honor to welcome our today's first distinguished and eminent speaker for this international conference. And I heartily extend a warm welcome to Professor Parthasarathy Mukherjee on behalf of Deshbandhu College and also on behalf of the Internal Quality Assurance Cell of Deshbandhu College. Sir, good morning and a very warm welcome to you. I'm also, I extend a warm welcome to our Principal, Professor Rajiv Agrawal, our Vice Principal, Professor Kamal Kumar Gupta, and all the faculty members of the Department of Chemistry and all the faculty members from across the world and students and research scholars. It is indeed my pleasure to formally introduce our today's distinguished speaker. Professor Parthasarathy Mukherjee is a professor of inorganic and physical chemistry at the Indian Institute of Science, Bangalore, and his main research fields include supramolecular and organometallic materials, organic nanostructures, molecular sensors, and catalysis in nanocages. Professor Parthasarathy Mukherjee is the Fellow of the Royal Society of England. He is also the Fellow of the Indian National Science Society. He did his PhD from the Indian Association for Cultivation of Sciences and his MSc from Jadavpur University. He has also been the postdoctoral fellow at two universities, University of Utah at the United States of America and the Göttingen University, Germany. He has been a professor in chemistry at the Indian Institute of Science, Bangalore since 2016. Earlier on, he was an associate professor over there from 2010 to 2015 and an assistant professor also over there from 2005 to 2009. He has also been the visiting scientist at the Universities of Heidelberg and University of Bonn. He has also been a visiting professor at the University of Oslo, Korea. And he has been the JSPS visiting scientist at the Kyoto University. He has been a member of the International Editorial Advisory Board and has been on the editorial board of a number of leading international journals like the Inorganic Chemistry of the American Chemical Society, Inorganic Chemistry Frontiers of the Royal Society of Chemistry, Inorganica Chemica Acta from the Elsevier Group, and the Chemical Records from the Wiley Groups. And he has also been an editor of the scientific reports of the Nature Springer Group. He has a number of awards and distinctions to his credit. He has been selected as one of the 75 scientists under 50 who have shaped India's today by the DST. He was elected the Fellow of the Indian Academy of Sciences in 2020. He was awarded the National Prize, Deshpande National Prize in Chemistry in 2018. He is also a recipient of the very, very prestigious Shanti Swarup Bhatnagar Award in Chemistry in 2016, which is the Indian equivalent of the Nobel Prize. He has been the Associate Editor of Inorganic Chemistry. He won the Chemical Research Society of India Bronze Medal in 2016. He was awarded the Fellow of the Royal Society of Chemistry in 2014. He was given the Young Scientist Award in Chemical Sciences, the Nasi Scopus Young Scientist Award in 2013, the Swarn Jayanti Fellowship by the Government of India in 2012, the Young Affiliate Fellowship, World Academy of Sciences Chivas, Trieste, Italy in 2011, the Microsoft Research Outstanding Young Faculty Award in 2009, the Indian National Science Academy Medal for the Young Scientist in 2008, the Young Associate Indian Academy of Sciences in 2008, and the Alexander von Humboldt Fellowship, Bonn, Germany in 2004. His teaching and research interests include basic and advanced inorganic chemistry, supramolecular chemistry, and has been actively involved in research on engineering molecules of defined shapes, sizes, supramolecular chemistry, organic cages, heterogeneous catalysis, cage-hosted nanoparticles for catalysis, molecular and polymer-based sensors for explosives, light harvesting materials, cage-based nanoenzymes. He has also mentored more than 18 PhD students, and currently 10 PhD students are working under him. He's also mentored 22 postdoctoral fellows, and he has also has another 4 postdoctoral fellows working under him. His total publications are 194, with total citations exceeding 12,026 and average citations per paper exceeding 61.67. And he has an H index of 61. So, I mean, this publication data is something that most people can only dream of. And we are really, really grateful, sir, that today you've taken out time from your busy schedule to deliver this invited lecture at the International Conference on Molecular Vessels. Sir, I welcome you and I invite you to now deliver your talk. Thank you.
May I share my screen now?
Yes, sir, you can.
Is it visible?
Yes, sir, it's visible.
Okay. Thank you, Dr. Saxena, for your kind words of introduction. And let me begin by thanking the organizers for the invitation. My group at Indian Institute of Science actually works on engineering molecular architectures of defined shapes and sizes and use the confined nanospace of such molecular architectures as molecular vessels to study chemistry. We generally try to see how the chemistry or behavior of molecules are different in a molecular vessels compared to their natural behavior in glass vessels or in organic or solvent or in aqueous medium. So we design such molecular architectures using general concept of geometry as well as using the general concept of coordination chemistry. Let me show you our design strategy. For example, from the general concept of coordination chemistry, we know that palladium and platinum in their +2 oxidation state always adopt square planar geometry. Square planar geometry means they have dsp2 hybridization. So initially free metal ion has 4 dsp2 hybrid empty orbitals. As all the 4 hybrid orbitals are available to bind with donors, we call it tetratopic acceptor, means 4 sites are available. Now, if we combine it with a bidentate chelating ligand like ethylene diamine, which is having 2 sites to bind with acceptor, then we can block 2 sites. So initially it was a tetratopic acceptor. Now it has become ditopic acceptor because it has 2 sites available to bind with donor now. Now the angle between them is 90 degrees. So we call it 90-degree ditopic acceptor. That is what coordination chemistry gives us this idea. Now let us use our general concept of geometry. If you imagine a molecular square, square has 90-degree corners and 4 linear arms. Now, if we use these as 90-degree corner, if we use this as a 90-degree corner, Then if we combine it with a linear donor, for example, 4,4-dipyridyl, it has lone pair of electron here to donate electron. This is another site through which it can donate electron. So, and the angle between these donor sides is 180 degrees. It is linear. Now, this is a bidentate donor because it has two sides to bind with acceptor, and it is a ditopic acceptor to bind with two donor sides. Okay, so if we mix them in 1:1 molecular ratio or molar ratio, then they will satisfy their coordination number. You see, this donor will satisfy coordination number because it has two donor sides, it has two acceptor sides. 1:1 molecular ratio, molar ratio is good enough. Now, if we mix them as per the general concept of geometry, this linear donor will occupy the sides of the square, and the corners of the square will be occupied by this 90-degree acceptor. So 4 acceptors, 4 donors will assemble together to give you a molecular square. Now, if we functionalize this building block with different functional groups, like if we introduce carboxylic acid here, there will be many carboxylic acids inside this pocket. So this pocket will be highly acidic. It can trap basic analytes. So if we have to separate any basic analytes or impurities from a mixture, we can easily use it as a trapping agent to absorb the basic analytes in its pocket and then separate. Okay, so we can use in this way for different purposes. I will show in next sections. So using the general concept of geometry, if we can design a building block which looks like a clip and then assemble it with a 180-degree building block, then 2 clips, 2 linear building blocks will assemble together to give you a rectangle. Similarly, 360-degree building blocks, 3 linear arms can combine by 3 + 3 self-assembly to give you a molecular triangle. Many 2-dimensional and 3-dimensional architectures can be designed in this way. If we use both the acceptor and donor ditopic and didentate, then resulting geometry will be a 2-dimensional architectures like hexagon, pentagon, square, rectangle, etc. But if one of the building blocks has more than 2 sides to bind with other building blocks, then the resulting geometry will be a 3-dimensional architecture. Now, in next part of my talk, I will show you how chemistry of photochromic compounds behave differently or is different from its chemistry in glass vessel. So this compound is known as pyropyran. And these 2 rings, pyros ring means they are not planar, they are almost orthogonal. So this is a non-planar molecule. The stable form of this molecule is this one, non-planar. Now this non-planar spiral compound will become planar conjugated compound upon irradiation with light. And because of this extended conjugation, it will be dark in color, either red or blue sometimes, but this will be almost colorless. So colorless form converts to its colored isomer, which is open in nature compared to the initial geometry. Now, as by applying light, irradiation with UV gives this one, this is called photochromic compound. Photochromic means, photo means light you apply, you change its color. Okay, so this photochromic compound or these kind of photochromic compounds are widely used for different purposes. For example, your eyeglass can be coated with photochromic compound. In presence of sunlight, in presence of UV light, it will become dark. So you can use it as goggles. But as soon as you get into room or in dark or in the evening time when no sunlight is present, it will become transparent. But for practical application, you need to have photochromic compounds that will show such color change in solid state. That means solid state, in solid state, such structural transformation needs to occur. But This is a non-planar molecule like this. It becomes planar after irradiation, means non-planar becomes planar like this. So for this structural orientation or change, you need to have enough space for this movement. But in solid state, molecules pack very close. So you do not have enough intermolecular space for this rotation. So solid state photochromism of these pyro compounds is very difficult to achieve. Now, these two isomers can be easily distinguished by UV because this colorless form, closed form, does not show any absorption in visible region. That means 400 to 600 nanometer region. But the colored one, it is highly colored. It shows strong absorption near 550. So this peak indicates that it is indeed in open form or this is in color. This one. So if you irradiate this solution in organic solvent, this stable compound will convert to this unstable, this form, but its stability is very short, very weak. It is called transient merocyanin form. This is pyroform. Its lifetime is just a few seconds. If you remove from UV source, immediately it converts to its colorless stable form. So you cannot stabilize this one. Not only that, if you heat this compound, it also converts to it. This is called thermochromism. By applying heat, you can change its color. Now, this is the general behavior of this pyropyran compound in glass vessel. If you take in a beaker or RB or in a test tube or in an enema tube, in organic solvent, irradiate with UV light, you see this one. Immediately it comes back. If you cool it down or just keep under room light without UV irradiation, immediately it will come back. Now, the question that we ask were, what will be the fate of such photochromic molecules in molecular vessels in confined nanospace? Is it possible to have such photochromism in solid state? To address these two questions, we actually designed a molecular barrel. Okay, so this is a tetratopic donor. This is one nitrogen donor through which it can donate lone pair. This is second, this is third, this is fourth. We still have two more pyridyl units with lone pairs, but these lone pairs cannot donate to acceptor because they are surrounded by bulky pyridyl ring. So this lone pair cannot approach an acceptor. Not only that, pyridyl groups are electron withdrawing group. They are in ortho position. So they will drag electron density from this nitrogen. So its tendency to donate electron is very weak. Now, this tetratopic donor or tetradentate donor looks like this, a panel with four corners having lone pair of electrons to donate. Now, when we assemble it with a 90-degree acceptor like this nitro and this nitro can be easily replaced. They are good living groups by these donor nitrogens. Now, in initial stage, when we mix them in DMSO, we got two different architectures. One is symmetrical barrel, another one is non-symmetrical barrel, which is assumed as if the symmetrical barrel breaks to form this one by sliding these two panels. When we added water and heated, we found only symmetrical barrel to form. And both the barrels were crystallized from this mixture and characterized by X-ray diffraction. You see, this barrel has large window at the top and bottom. And it has large confined pocket, which is of almost 2 nanometer diameter. That means diameter is few nanometers and confined pocket is quite large, few nanometer dimension, large pocket. Now, we wanted to use that barrel as a molecular vessel instead of using glass vessel. So this one is highly soluble in water. This is the aqueous solution of barrel. Now you take this pyro compound, it is commercially available. You can buy also, you can prepare also. This one is non-planar. This is the stable form. It is almost colorless. If you add this solid, it is not soluble in water. But as soon as you add it to aqueous solution of barrel, it is soluble quickly in a few seconds. That means if you take simple water and this one, it is never soluble. But if you take water plus our barrel, then add this one, then it is easily soluble. It goes into confined hydrophobic pocket. This pocket is hydrophobic because it is surrounded by aromatic building blocks, but overall this is highly soluble in water. So this confined hydrophobic pocket accepts this molecule. Not only that, immediately we could see color change. And this happened in room temperature without heating, under visible light, without applying UV light. As I mentioned initially, that this molecule is the stable form under visible light at room temperature. You have to heat it with heating, or you have to irradiate with UV light to get the open form. Now we check the UV of this solution. Why is it green? We found a strong absorption at 590 nanometers. So this indicates that molecule, this one has been converted to open form now. That means it is no more existing in this form, but this transformation has happened at room temperature and under visible light. It is quite surprising. Not only that, even if you keep this solution, green solution, for several days, months, it will remain green. That means inside this molecule, you can stabilize this transient form. And this was also proved by NMR that indeed this molecule exists in this open form in presence of this pair. This is quite surprising. For last 50 years, physical chemists have seen that this pyro compound is the stable form and merocyan in open form is unstable. Its lifetime is just few seconds. But in presence of this barrel, at room temperature, we could convert this one. Not only that, the stable form is this open form, not the closed form. Now, is it only restricted to one barrel or one photochromic compound? Now, bromo has been substituted by nitro. It's a different photochromic compound. In that case also, when we added our photochromic compound to this barrel solution, you see it becomes red. And UV and NMR prove that this red form is due to the open form of this compound, not due to this compound. Okay, this is colorless. Now, you see it becomes red, but if you just shake it with chloroform for a few minutes, you see chloroform is coming at the bottom, and this blue compound, that means open form, comes to the chloroform medium because it is highly soluble in organic solvent. As soon as it comes to the organic medium, in a few minutes, it becomes colorless open form. That means in organic medium, its stable form is this one, which is known for several decades. But when it was in molecular vessel, it remains in open form. You see, this is the color change. Now, to see whether it is only restricted to one barrel or it can show different behavior in other barrels also, we designed another barrel using tetraimidazole instead of tetrapyridyl ligand. With this case gave us wider barrel. So this diameter is actually even wider in this case because this ligand is more wider. In this case also, both nitro and bromospiropyran showed similar behavior. That means at room temperature, we could stabilize this one for several months, open form, and without UV light. Now, the overall story is a molecular vessel which is colorless in aqueous medium. Now, if you add this colorless compound, it becomes blue or red. Now, this compound exists in open form without applying UV light, without applying high temperature. You can easily convert to this one in presence of barrel. But normally, without barrel, it will never convert to this open form. It will remain in this form. You have to irradiate with UV light or heat at high temperature to get this form, but this form is unstable. As soon as you cool down or remove from UV source, it becomes this form. But in this case, this color solution becomes colorless only if you heat with UV light or UV light, irradiate with UV light or heat it at high temperature. Then the solid compound will come out. Again, if you keep it for a few minutes, it will dissolve and give you blue solution. So it is showing reverse thermochromism and reverse photochromism in presence of molecular vessel. Now the second question was, is it possible to have solid state photochromism? Now our barrel has wide open pocket in its molecular cavity. Now this is the spiro compound, which is light yellow or cream color. This is our molecular barrel. If you just mix them and grind for few minutes, you can see drastic color change. You see, if you irradiate with UV light for few hours, it will become even intense. That means spiro compound becomes merocyanine. After 10 hours, you can see the color change. Okay, so in solid state, you can see photochromism in presence of barrel. But if you simply take this one and keep under UV light for 2 hours or 10 hours, you won't see any color change. That means barrel promotes this color change or conversion to open form in presence of UV light. Now we dissolve this one in organic solvent like hexane, made a solution because spiropyran is soluble in hexane. This is barrel, we made a solution in water. To that aqueous medium, we just dipped a filter paper and then took out and then dried using heat gun. Now, this dried filter paper is having solid barrel because it was dipped in barrel solution. Now you removed water by heating. So barrel is there. So your filter paper is coated with solid barrel. Now hexane solution of this spiro compound was used as an ink. And then we wrote something. Nothing is visible normally because in solid state photochromism is not easy. But we irradiated with UV light. Under sunlight, we kept for a few minutes. We can see all the writings. They are visible. Even if you remove from UV light, you can see this visible color change. And this color will remain for several weeks without any change. Now, if you dip this one in an organic solvent like chloroform or dichloromethane, then this organic compound, that means the open form of our photochromic compound, will be highly soluble in water. So that will be washed out. But your filter paper still has molecular barrel, which is not soluble in organic solvent, which is soluble only in water. Now that filter paper can be again used for writing this. So you can reuse this filter paper as rewritable pad, magic pad. So far I have shown you that chemistry of photochromic compounds can be completely opposite to what chemists have seen for last 50 years in glass vessel. We have also used other photochromic compounds to study unusual behavior. I'm not going to discuss this result, just I wanted to show you different kind of results or different kind of chemistry that we actually do in our laboratory. So next part will be chemical reactions in molecular vessels or in confined nanospace. The question that comes to our mind first is, why do we need confined space for chemical reaction? We are comfortable in doing chemical reactions in glass vessels, round bottom flask, beaker, conical flask, etc. But the concept of chemical reaction in confined nanospace is not new. It is nature's gifted idea. If you imagine your many chemical reactions that happen, enzymatic reactions happen inside the confined pocket surrounded by enzyme molecules. And such reactions are so specific that they happen only inside confined molecular pocket surrounded by enzyme. Same reactions will not happen outside enzymes pocket. This is marvelous, nature's gifted result. Now, chemists actually try to mimic such nature's gifted idea to design artificial molecular enzyme or molecular vessel. Now let us see the physical chemistry aspects of doing chemical reaction in confined space. Why confined space? Or what are the advantages of chemical reactions in confined space? Let us assume a reaction of 4 different reactants A, B, C, D, and they condense to form one product P. That means 4 molecules convert to one molecule. In this case, what will happen to the entropy? it will be an entropy loss process. In a glass vessels, you are doing this reaction in presence of, suppose, 10 milliliter of organic solvent or aqueous medium. So this is an entropy loss process. That means final entropy is less than the initial entropy. So S2 minus S1, that means del S is negative. We know del G is equal to del H minus T del S. If del S is negative, del G will be positive. So thermodynamically, reaction will not be favorable. So entrophically, reactions are not favored in this kind of reactions. Now, instead of taking them in a big conical flask, imagine you take them in a confined molecular vessel, where molecules are tightly packed, they cannot freely move. So initial entropy of the reactants you are reducing. Rotational and translational degree of freedom will be restricted in confined pocket. They cannot move freely. So initially in flask, entropy was very high because you had 10 milliliter volume. Now you have a few nanometer volume. So entropy will be less. Reactants entropy you have reduced. That means S1 you have reduced. S2, after formation of product you are taking out. That means entropy will be like previous entropy of the product. So S2 minus S1 now is less negative. Earlier it was very high negative. Now it is less negative. So entropically you can make reaction more favorable. Not only that, if you take this reactant, suppose 1 milligram reactant in 10 milliliter solvent versus 1 milligram reactant in 1 nanometer flask. Molecular flask means very tiny, you cannot see. 1 nanometer suppose. Then what will happen to the effective concentration? Volume is reduced drastically. So entropy concentration will be very high. So as per Les Atelier's principle, N2 plus 3H2 is equal to 2NH3. If you actually increase concentration of the reactants, what will happen? Product formation will be more. Here effective concentration is increasing, so reactivity will be high. You can expect more product formation. Not only that, if you can take out this product from the pocket after formation, N2 plus 3H2 is equal to 2NH3. If you remove NH3 from the system, more product formation is expected. So if you take out it from the pocket will be free, reaction will be facile in forward direction. Now, if you design this vessel using appropriate building block that can stabilize the transition state of a reaction. You see, reactant you have to activate with energy. This is transition state. Then product formation will happen. Either it will have lower energy or higher energy than reactant. But this activation energy will be less if you can stabilize the transition state. Stabilize means you are reducing the energy. So activation energy will be less. Reaction will proceed through a different pathway. Reaction will be catalytic in nature. So in this way, you can actually get catalytic turnover. Small amount of this vessel you can use. It will act as catalyst and give you large amount of product. Now, there are many other advantages I will show you. This is a molecular cage, which is represented by this closed cycle for simplicity. This is soluble in water. This reaction is known in textbook. If you take acinaptine in organic solvent, irradiate with UV light, then this double bond and this double bond will cyclize. It is called 2 pi electron plus 2 pi electron cyclization to give you cyclobutane ring. Both syn isomer and anti-isomers form in almost equal amount. So to separate them, you need to do column chromatography. Their physical properties are same, very difficult to separate. So it is extensive. It will take extensive time as well as it is expensive. Now, when Fujita's group took these two molecules inside this vessel, where molecules cannot move freely, and they orient in a particular fashion like this parallel fashion. They are not orient in opposite fashion like this to have this product. So in this case, when they irradiated, they only found the formation of this product, no anti-product, no separation was required. But the problem of this approach is following. this product is bigger than the reactant. Reactant can easily pass through this narrow window and can go and sit inside the pocket. But once the bigger product is formed, it cannot come out. So it is basically caged inside a cage. So you have to break this cage. That means you are destroying this palladium cage. It is again expensive. So it is called stoichiometric reaction. So one molecule of cage can form only one molecule of product. Now the question that we asked is, can we take out from the cage automatically after formation? If it comes out, then this cage will be free. It will take another molecule, form this kind of product, and it will be a continuous cycle. So if you use 2 or 5 molecules of cage, it can produce 5 lakh molecules of product. So our idea is to design molecular barrel, because barrel has large window, 2 windows, bottom and top sides. And if they are soluble in water, then you can perform reaction using reactants that are not soluble in water. They can go to this hydrophobic pocket. And if product can form, it will come out, because pocket size is identical to window size, because barrels are like this. So they will have open windows same as pocket size. So with this idea, we develop several molecular barrels and use them as molecular vessel to study chemistry of different kind of molecules, separation, catalysis, etc. I will show you one example which is not water soluble. Now, this urea in solid state from intermolecular hydrogen. So in solid state, if you take urea as catalyst to perform reaction, it is not easy, because in solid state, these hydrogens are not free. They are already hydrogen bonded. But this free form is active mode that can activate compound like nitroolefin. If you add, it will form hydrogen bond, and then this will activate this carbon to perform chemical reaction. But in dilute solution, it will break to form this open monomeric form, which is active form, and then can catalyze. But most of the organocatalytic reactions are done in organic solvent. But in aqueous medium, people don't do. We will see. Moreover, such reactions are homogeneous. That means after the reaction, catalyst cannot be separated and reused. Now, we thought, can we reuse and check what will happen if we use urea-based building block, dipyridyl unit. This is one and this is one, and we have 2 urea moieties represented by these red dots, and these balls represent pyridyl nitrogen donor. When we treated with, we checked this X-ray and found that all the urea NH photons are involved in hydrogen bonding. So if you take this one as catalyst, it does not show any catalytic activity. Now we treated it with 90-degree acceptor, which is having 2 open sides. These 2 sides can be easily replaced by pyridyl. So they are available, and this is amine. We got a molecular triangle. But this also does not show a catalytic activity, because you see this triangle can come on top of another triangle, that will come on top of another triangle like this. And in that case, these NH photons will form hydrogen bond with the CO moiety of another triangle. So they are not free. So they are stacked like this. So we thought, if we got this triangle using this background, we thought, if we do not have amine on each palladium, what will happen? Each palladium will have 2 sites available free. Now, if we can bring 2 triangles close enough, but we cannot talk to the triangle, they cannot understand our language. So we need to have a binder that can bind here as well as here. That means we can stitch 2 triangles here also, here also. By this linker, then we can create a 3-dimensional pocket like this. But we have to keep in mind, if these 2 triangles come close, then again this urea and this urea moiety will form hydrogen bond. So urea moieties will not be free to activate your reactant. So we have to choose a building block which will keep them away at least 6 angstrom. So this pyridyl nitrogen, if it can bind here, this will bind here, and then the urea will be at least 5 to 6 angstrom away, so they will not form hydrogen bond. With this hope, we designed this and treated with this. So instead of palladium with ethylene diamine here, we use simple palladium nitrate. So no diamine on each palladium. And in this case, we got this kind of prismatic structure where these NH protons are free, you see X-ray sword, and they are projected inward and they are not hydrogen bonded. So you have a three-dimensional pocket, large open windows, and pocket is surrounded by many urea protons. So they can bind with substrate which can form hydrogen bond with urea. We checked like this one. This one is colored in organic solvent. It gives you yellow solution. To that, you add our barrel or this prism. Prism is not soluble. You see solid, colorless solid. After 2 hours become yellow and solution becomes colorless. That means it absorbs this one and then become yellow. So in this case, and then we have performed this reaction. We got successful result in all the cases. I'm not going to explain the mechanism. Our barrel has large open window that actually releases the product after formation and then can this. So how much time do I have? 10 minutes?
So 5 minutes more.
Okay, I'll finish in 5 minutes. So in this part, I will show you separation of two components. You see, anthracene crude oil is the major source of phenanthrene, which is widely used in industry. Okay, but separation of phenanthrene from its isomer anthracene, both of them have similar physical and chemical behavior is very difficult. So that cost actually huge. So we thought we can use a flexible ligand with this 90-degree acceptor. In this case, we got a molecular boat. And this molecular boat can encapsulate phenanthrene as well as anthracene. We have studied binding, checked binding constant. We found phenanthrene binds stronger than anthracene. And as it has better binding affinity compared to anthracene, we thought, can we separate them by using it as an extracting agent? It is highly soluble in water. Anthracene, phenanthrene, they are not soluble in water. We took a mixture of anthracene and phenanthrene. Okay, these are all crystal structure characterization. They showed that phenanthrene, pyrene can bind inside the pocket. We have proved, and then by UV and NMR study, we have checked binding constant. Phenanthrene case binding constant is almost, you see, 20 times or 30 times more. So this one, then we took aqueous, simple solid phenanthrene and anthracene mixture, and then aqueous solution of our boat. Okay, so phenanthrene is highly soluble, anthracene is weakly soluble inside this molecular boat. Binding affinity is more for phenanthrene. So phenanthrene goes in aqueous medium because in aqueous medium we have barrel or boat. Anthracene is not soluble. You can simply filter. So anthracene can be separated. Now, how can we extract this phenanthrene? You add chloroform. In chloroform medium, and phenanthrene will come. Aqueous medium has steel barrel, boat that you can again use for this kind of separation. And we checked 5 cycle, and you see the performance is very high, almost 99% pure phenanthrene you can isolate from a mixture of anthracene and phenanthrene. So this has been recently accepted in JCS. And in the last part, 2 minutes I will take, we also use organic molecular vessels. For example, if we take this amine and then aldehyde, we can get imine, and this kind of cages, imine bonds are sensitive, and then we reduce them to amine to create 3-dimensional organic cage. Okay, different sizes. And the confined space we use for nucleation of nanoparticle. For example, you have NH. If you add palladium or gold like that, they can go inside and bind. And metal ions are inside this pocket. Now you reduce this palladium 2 with a reducing agent that will form palladium 0. Palladium 0 atoms will combine to form palladium nanoparticle inside this confined cage. Then nanoparticle size will not be bigger than the size of this pocket. Okay, in that way we can actually control the size. And we have done gold, palladium, silver nanoparticle less than 2 nanometers, and then use them for different catalytic reaction. For example, I will show you one catalytic reaction using this cage. We prepared gold nanoparticle and simple reaction, nitro to azo reaction in 12th standard you have studied that it gives azoxy, azo, hydrazobenzene, etc. mixture, but it doesn't give selectively azo compound. But using our cage supported nanoparticle, gold nanoparticle, which are very tiny, less than 2 nanometer dimension. So in 2 hours time, we selectively got almost 100% product in all the cases. No byproduct was formed. And this is widely used in industry because azo compounds are used for making colors in dye industry. And simply from nitro to azo, you can prepare without using, without any byproduct. So that's all. We have also reused this catalyst many times, 5 times, without much loss of catalytic activity. So we have checked the stability of the catalyst after 5 cycles. It remains stable, and also studied mechanism. I'm not going to discuss again this one. So I'll stop here. So in the conclusion, as I mentioned, we are fascinated in designing molecular barrels which are soluble in water, and they have wide pockets. So chemicals can go and come out inside also. So we use such molecular vessels to study chemistry of different class of compounds. We use them for performing catalytic reactions so that product can come out after formation. Stereoselective product can be formed. Moreover, we can also use them for separation of isomers from aqueous medium using water insoluble substrates. Also, we also work on organic cage compounds, use them again as molecular vessels to form tiny metal nanoparticles, and then use that cage supported nanoparticle as catalyst for various chemical transformation in heterogeneous catalytic manner. So finally, I'd like to acknowledge my students who have done the work. Pradeep Hauladar, who is an Humboldt fellow now in Germany, who worked on molecular vessel for chemistry of photochromic compound. Ganeswar Mondal, who is now an assistant professor of a central university, he worked on organic cage. Rupak Saha also worked on photochromic compound. Arpita worked on the last part, that means separation of isomers using molecular board. And Ennio Jangrando, who helped us in collecting synchrotron X-ray data for some of our system, SCRB for financial support. And thank you very much for your kind attention.
Thank you so much, sir. And we are really, really grateful for such a lucid, such a simple and informative talk. And especially the way you presented all these concepts, even though I am a student of physics, but even I could follow quite a large part of your talk. So that is really, really very critical. And I'm really grateful to you, sir, on behalf of Department of Chemistry and on behalf of Deshbandhu College for giving such a wonderful talk. As a token of our gratitude and appreciation for you to take out time for us, I would now request that please accept an e-certificate and an e-memento from our side, from the Department of Chemistry, Deshbandhu College, and from Deshbandhu College.
Please, sir, stop your sharing, sir. Dr. Partha, sir.
Yeah.
Yes, sir.
Thank you.
So, I once again thank you very much, sir, for sparing time and joining us and delivering such a wonderful lecture on molecular vessels. And now I would request Dr. Aparna, the chairperson, co-chairperson of this session, to take over the proceedings. Over to you, Aparna.
Thank you, Dr. Aditya Saxena. With your permission, can I start the next session, Dr. Jha?
Sure.
Thank you, Dr. Aditya Saxena. With your permission, can I start the next session, Dr. Jha?
Sure.
Thank you, Dr. Aditya Saxena. With your permission, can I start the next session, Dr. Jha? Please unmute yourself.
Yes, you can start.
I think we are all audible. Uh, Thank you, sir. It is indeed a pride moment for us at Deshbandhu College to welcome and introduce the speaker, Professor Dr. Amitabh Jha, in this session of Day 2 of the International Conference on Recent Advancements in Chemical Sciences. Dr. Jha, who is alumni of Department of Chemistry, University of Delhi, which is also celebrating its centenary year. Dr. Jha is currently a full professor at Acadia University, Nova Scotia, Canada. He obtained his master's degree from University of Delhi and then doctorate degree also from the Department of Chemistry, University of Delhi, under the supervision of Professor V.S. Parmar. And after a brief stint with Ranbaxy Laboratories, New Delhi, he moved to Canada, wherein he was associated with College of Pharmacy, University of Saskatchewan, and at Alviva Biopharmaceuticals. And then subsequently, he joined Department of Chemistry at Acadia University in the year 2002. Since then, he has worked in different capacities in the same institutions and is a recipient of numerous honors, awards, and has 3 patents to his credit. He is also on editorial boards of several national and international journals and has more than 80 publications to his credit. And his area of interest is medicinal and synthetic chemistry. He has also mentored numerous undergraduate, postgraduate students and is supervising doctoral students also. So without taking much time, may I now invite the esteemed speaker to address the participants. Over to Dr. Jha.
Thank you.
You may start, yes.
So are you able to see my screen?
Yeah, it's visible.
Okay. Hello, everyone. And I would like to thank the organizer to give me the opportunity to speak at this conference. I'm really delighted, and as Delhi University is my alma mater, it's always a happy moment to actually interact with students of Delhi University and colleagues at Delhi University. As Aparna mentioned that, I work in the area of medicinal chemistry and synthetic chemistry. Today, my talk is going to be on medicinal chemistry, and I'll be sharing some of our results on selective estrogen receptor modulators. So as Aparna mentioned, we work in the area of medicinal chemistry. And within medicinal chemistry, we are working on selective estrogen receptor modulators, topoisomerase inhibitors, analogs of curcumin. Many of you might know curcumin is the active ingredient found in the spice turmeric, or haldi as it is called in India. So we have done quite a bit of work on that. In the synthetic organic chemistry, I work with different types of reactive intermediates like quinone methides, N-acetyluminium ions, and donor acceptor cyclopropane chemistry. And there are some of the results from our lab, which we published on synthetic chemistry in various journals. In the area of topoisomerase inhibitor, here are some of the compounds where we have found interesting and remarkable anti-cancer activity. Well, topoisomerase is actually an enzyme that helps in unwinding DNA during the cell division process. So if you are able to stop the activity of topoisomerase, eventually the cell death will occur. So that's how these compounds show their activity. Curcumin analogs, the top structure that you see is the structure of curcumin, and we have in our lab made many series of compounds resembling the structure of curcumin and looked at their biological properties and matched it with curcumin. And many of our compounds had better activity compared to curcumin. And as my focus is on cancer, we looked at the anti-cancer activity of these compounds. Splitomycin is histone deacetylase inhibitor, which has application in cancer as well as several other diseases. And we have found ways to make splitomycin compounds and are in the process of testing their activity. My focus of the lecture today is going to be on SERMs, and I have structure of various types of SERMs that we have made, and I will be talking about these compounds today. So first, what are SERMs? And before SERM, I think we need to understand what are estrogens. Probably everyone in the audience knows what estrogens are. Estrogens are, they're popularly known as female sex hormones. However, they do more than that. The function of estrogen is actually much more than, just the like having effect on the female sexual characteristics. So generally speaking, I have the structure of three estrogen molecules. They're collectively called estrogens. They are, as you can see, they're steroidal in structure. They're produced and secreted by ovaries. They are hormones, and hormones, as you probably know, are signaling molecules. They stimulate the development and maintenance of female characteristic and sexual reproduction. They travel through bloodstream and they interact with cells in a variety of target tissues. In other words, estrogens play a role in different type of tissues within the body, and so basically, in this picture here, I'm trying to highlight where estrogen plays a role. So estrogen plays a role in brain, heart, breast, liver, uterus, and bone. In terms of the activity, estradiol is the most active estrogen molecule. The estriol is the least active, and estrone is kind of, somewhere in between. Now, how is estrogen responsible and how is it related to cancer? So estrogen is a signaling molecule or a hormone, and basically it goes and binds with estrogen receptor in the tissues where estrogen receptors are found. Estrogen receptors are nuclear receptors. They're found inside the nucleus. So basically, when estrogen binds with estrogen receptor, there is a few things happen. So when estrogen binds with estrogen receptor, basically, it comes in contact with several coactivators, which then binds with the gene, and gene gets activated. And after the activation of gene, messenger RNA is created, and the messenger RNA leads to synthesis of several proteins, and these proteins then lead to change in the cell behavior. So depending upon in which tissue this is happening, the corresponding type of activity is triggered. For example, if it is happening in brain, it leads to neuroprotection. If it is happening in bloodstream, it has effect on the blood lipid profile. If it is happening in breast tissue, it leads to proliferation of breast cells. If it is happening in uterus, then there is proliferation of uterine cells. So likewise, wherever estrogen receptors are found, estrogen molecule leads to change in the cell behavior there through this process. So as I already mentioned, increased proliferation in breast and uterus, it decreases the LDL, which is the bad cholesterol, and it raises the HDL in liver, which is the good cholesterol. In heart, it does cardioprotection. In bone, it leads to bone maturation and makes the bone stronger. In brain, it leads to neuroprotection. So a lot of function is basically, this molecule estrogen does. My focus is on what estrogen does in the breast cells, okay? And basically, if you look at the biosynthesis of estrogen molecule, they are basically converted from male sex hormone to this estrogen molecules by the enzyme aromatase. And if you pay attention to the structure, you will notice that in estrogens, the first ring is aromatic, whereas in the male sex hormones, these rings are not aromatic. So in breast cancer therapy, if a patient has breast cancer, how you can stop breast cancer or how you can mitigate it. Basically, in the presence of estradiol, the breast cancer cells multiply faster. So your idea would be to actually somehow make a compound which is anti-estrogenic that stops the activity of estrogen. That would have a beneficial effect. Or basically you do not let the formation of estrogen at all. And in that case, you would be making aromatase inhibitors. So if you inhibit the enzyme aromatase, that will lead to no formation of estrogen. And if estrogen is not forming, then the proliferation of cell will stop in the breast cells. So what happens with the estrogen receptor and breast cancer? So generally speaking, the job of estrogen is to activate the gene inside the nucleus of the normal breast cells, and it leads to proliferation of normal breast cells. However, let us say there is a mistake in DNA duplication and it has led to formation of a cancerous cell. This cancerous cell starts to multiply even faster than the normal breast cells, and that forms the tumor. So estrogen, which actually has, the good activity in the body, if there is a mistake in the DNA duplication and somehow the breast cells have become malignant or cancerous, then in presence of estrogen, they are going to multiply really fast. So basically, if you are able to stop the effect of estrogen, that would be one of the ways to actually stop breast cancer. There are two types of estrogen receptor alpha and beta, and they are often co-expressed. That means in almost all the cells, wherever estrogen receptors are found, both alpha and beta receptors are found. However, it is the estrogen receptor alpha which is the prognostic marker for breast cancer. So if a molecule is able to inhibit the attachment of estrogen at the estrogen receptor alpha, it will have the beneficial effect in breast cancer. So in medicinal chemistry, molecules which actually play or they interfere with the activity of estrogen, they are called selective estrogen receptor modulators. Now, this is an interesting term, modulator, because normally in medicinal chemistry, you would hear the terms like agonist or antagonist or activator or inhibitor. Modulator is basically a different type of word, which it simply means that it is able to do some change in the behavior of, in the environment wherever it is acting. So SERMs, these modulators, they are not pure agonists or antagonists. Their modulatory activity may be different at different locations. A SERM molecule can be agonist at one cell or at one location, and the same molecule can act as an antagonist at a different location. So just to give you an example, let us say you have a SERM molecule and it goes and binds with the estrogen receptor at breast cell, and it is able to completely stop the activity of estrogen there, and there is no breast cell for proliferation. This would mean that this SERM molecule is acting as an antagonist in the breast cell. And the same molecule, when it is received at the estrogen receptor in uterine cell, it can actually activate the uterine receptor, and it will lead to uterine cell proliferation in the uterus. So same molecule is antagonist in breast cell, and it is an agonist in the uterine cells. So this happens often with these types of compounds, and therefore they are called modulators. and not either agonist or antagonist or inhibitor or activator, because they do the job of both. So now let's look at some of the clinical examples of estrogen receptor modulators. Tamoxifen is one of the gold standard, and it has been known for a long time, and it is widely used for breast cancer. So this molecule is a SERM. The beneficial effects of tamoxifen is that it reduces breast cancer risk. It lowers LDL cholesterol, so the bad cholesterol is lowered, and it strengthens the bones. So tamoxifen is also used in case of osteoporosis. But the bad effect of estrogen is that it increases uterine cancer risk. So since it's a modulator, it acts as an agonist as an antagonist in the breast cancer, but as an agonist in the uterine cells. It also leads to increased blood clot risk. So, of course, there is a need to actually develop a compound that has less bad effects. So, one of the more recent compound is raloxifene, and it was initially introduced for osteoporosis, so it definitely strengthens bones. It was also found to reduce breast cancer risk, and in fact, it also worked more or less like an antagonist at the uterine cells. However, it also had some bad effects. For example, the blood clot risk did not go away, and there was no reduction in non-invasive breast cancer. So, I mean, even if you look at other SERMs which are currently being developed, none of them have, entirely all the good effects and no bad effects. However, continually scientists are able to achieve some improvement from the previous molecule, and same was the idea when I started, or when we started this work in my lab. So to actually understand how these molecules work, let us look at how estrogen binds at the estrogen receptor. So this one is a cartoon of the 3D model of estrogen inside the binding pocket of estrogen receptor alpha. And as you can notice that one of the hydroxy group of the estrogen molecule is forming a hydrate glutamic acid and arginine triad at the bottom side, and it also forms a hydrogen bonding with the hydroxy group at the other end of the estrogen molecule with histidine 524. So this is how this molecule fits inside the estrogen receptor and does the binding. Now, as you know, that raloxifene is a estrogen receptor, it's a selective estrogen receptor modulator. It was also co-crystallized with estrogen receptor, and this is the binding that was found with the raloxifene molecule. So raloxifene molecule also has, the 2-hydroxy group, which bind exactly at the same position where the hydroxys of estrogen binds with the estrogen receptor. In addition to that, the side chain, which is actually shown in the side chain, which is shown right here, this side chain, there is an additional binding with the nitrogen with aspartic 351. So there is a salt bridge that forms over there. So there is an additional site, and this additional site of binding is responsible for the desirable properties in the molecule. So in a lot of estrogen molecules, or in a lot of SERM molecule, selective estrogen receptor modulator, you will see that they contain a side chain similar to what is shown here. And even the compounds that we have made or synthesized contain the side chains. So let's look at, and I'm going to actually share with you a different series of compounds that we have made with the estrogen receptor alpha, with a hope that they are going to bind with estrogen receptor alpha and they are going to behave like SERM. So the first series of compound, as you can see on the slide, is the designed SERMs. So again, on the two sides, one side you have the estradiol, which has the 2 hydroxy groups, and raloxifene that also has 2 hydroxy groups and has the side chain in it. So the molecules that I had designed also contain 2 hydroxy group mimicking the estradiol hydroxy groups. There is another hydroxy which is kind of, it may have a role to play or it may not have a role to play, but then the molecule also contains the side chain similar to what is found on raloxifene. We synthesized, we wanted to synthesize the molecule, but what we found is that to actually make this designed compound is because of the 2 hydroxy group on the naphthol ring, synthesis of the molecule at one particular, to make this desired compound was relatively harder, not impossible, but harder. So what we thought, why not make the molecule that is easier to synthesize? And that molecule would be the one shown on the bottom right-hand side, where one of the hydroxy group is missing at position 6 of the naphthol ring. Now, this hydroxy group missing, it did not bother me much because if you look at the structure of tamoxifen, it does not contain any hydroxy group. However, when it goes inside the body, it gets oxidized and a phenolic group is formed, and that phenolic group helps in the binding at the right spot of the estrogen receptor. So with that hope, we synthesize these compounds. And if you look at the structure, it's basically a very simple chemistry. You can use manic reaction to make compounds like this. So, and manic reaction is a multi-component reaction. You react naphthol with a non-enolizable aldehyde and a secondary amine, and the reaction works really easily. In microwave, you can do the reaction in a minute. If you want to do it under conventional condition in like overnight, reflux in ethanol produces the molecule. So we made these series 2 molecules using this chemistry where we varied the side chain as A, B, C, D, E, and F. So, and in fact, the selection of these 6 different side chain was dependent on the commercial availability of the corresponding chloroamine compound shown right here. Okay, so these chloroamine compounds were commercially available, so we just did the reaction of this chloroamine with the para-hydroxybenzaldehyde, simple displacement reaction to form the aldehyde with the right side chain that we wanted to introduce. And using this particular aldehyde in the manic reaction shown here, it led to formation of series 2 compounds. There is one chiral center, and the chiral center is shown with the wavy bond there. So on that chiral center, we actually did some resolution studies, which we published in Journal of Molecular Catalysis. But I'm going to skip that. I'm going to talk about the biological activity. So here is the cytotoxicity data for the compounds I made. And as you can see, it is compared with the reference drug tamoxifen. So tamoxifen shows its activity, the IC50 value against the breast cancer cell line MCF7 at 31 micromolar concentration. And almost all compound except the one having morpholino group in the side chain showed almost identical activity to tamoxifen. So they were almost equipotent. So this basically gave us, some boost saying that, okay, we are on the right track. And maybe if we do further design of molecules, we might get better activity. Again, so with we continued our design. So again, I'm showing you our series 2 molecules. In the middle, you have the estradiol and raloxifene on the side. So basically, one series of compound we synthesized, we called it series 3, and series 3 was structured like this, where again, we tried to mimic one of the hydroxy group at almost the same position as found in estradiol. The other series of molecule had one carbon extra, and I showed you the superimposed structure. Again, these superimposed structure, whether they are going to be exactly similar under, will they have the same conformation like estradiol is hard to say. However, the open chain molecule has a larger degree of freedom, and it is possible that it can actually, fit. There could be an induced fitting at the binding site. And the series 5 molecule had structure like this, where similar to raloxifene, I had the naphthalene ring with, at position 2, we introduced a heterocyclic nitrogen containing compound, and at the 4th position of that, we had the hydroxy. And all of these compound has the side chain as required for the synthesis of SERMs. So we synthesize these molecules, and I'm going to quickly go over the chemistry of synthesis of these compounds.
I'm sorry to interrupt. The slides are not moving.
The slides are not moving.
Not.
So has it not been moving from the beginning?
It's there since number 10. After 10, it didn't move, perhaps.
Okay, let me.
The smaller
Okay, so
one are changing. The numbers are changing on the smaller one on right side. But the bigger one, it's there only, static.
One second, let me see if, so where is the share screen?
Share screen would be at the bottom of the Zoom Meet. One in green after chat. Control panel of Zoom.
Okay, so do you have this on the full screen now?
Yes.
Okay, so let me see now if it's working. So now, do you see, is it moving now?
Yes.
Yes, sir. Yes, sir. Moving, sir.
Okay, good.
Now
Thank
it's moving.
Thank you. So again, I guess I have to kind of go through over these slides again. So the design of compound, the structure in the middle is our design SERMs, and on one side I have estradiol, and the other side I have raloxifene. And basically, the similarity, as you can see on the design SERM, I tried to place 2 hydroxy groups mimicking the hydroxy of estradiol, and also include the side chain. And the molecule, can be synthesized by doing Mannich reaction. Just for the simplicity, we decided to make the molecule shown at this bottom right, which has 1 hydroxy less, because it was much easier to synthesize. So we made these compounds using, the Mannich chemistry. So in Mannich chemistry, phenol, aldehyde, and amine can give rise to a product like this. And if we use, in place of the aldehyde, this aldehyde containing the amine side chain, that would give us the compound that we wanted to make. So we did that synthesis using simple SN2 reaction between para-hydroxy benzaldehyde and chloroamine, and these chloroamines were commercially available. And that led to 6 compound in series 2. We did some resolution studies which we published, so that's not much of a consequence for the medicinal work. So I'm just going to talk about the cytotoxicity data. And for the cytotoxicity data, tamoxifen showed the biological activity at 31 micromolar concentration against MCF7 breast cancer cell line. And our series 2 compounds, 5 of them were almost equipotent to tamoxifen. Only one compound with a morpholino group on the side chain actually showed, it was inactive. So we were actually, pretty happy with these results, and we went on to synthesize, to actually design more compounds. And as you can see now, I have the series 2 compound on one side and raloxifene on the other side. And the new design, I'm going to show you with the overlaid structure on estradiol as well as on raloxifene, and then see how they are similar. So this is my series 3 compounds. Series 4 compound, the difference between series 3 and series 4 is that series 4 has an extra CH2, and the series 5 compound was similar to raloxifene. So we wanted to make all these 3 series of compounds. So before we started the synthesis, we wanted to actually see whether these compounds are going to actually bind at the estrogen receptor binding spot. And for that, we did some in silico work. We did some computer modeling, and in the computer modeling, we were able to see that the triad at which one of the hydroxy group was binding, our molecule was fitting there, and the other hydroxy was binding with the histidine on the other side, and the side chain was actually binding to the aspartic acid. So basically, it was behaving just like a raloxifene behaved under the co-crystallized structure with the estrogen receptor alpha. We also did a superimposed structure of our design molecule, a representative design molecule with raloxifene. And as you can see, the purple is the raloxifene molecule and blue is our design molecule, and their conformation are pretty similar. So we started synthesizing these compounds. So as I told you earlier that, there were 5 amino halides that were commercially available. 6 of them were commercially available, but we wanted to make more. So we actually made certain more amine substituted aldehyde for which we had to do the reaction from scratch. So we basically started with para-hydroxybenzaldehyde, reacted with dibromoethane, and by controlling the stoichiometry, we actually formed mono alkylated para-hydroxybenzaldehyde, and then using the bromo group, we displaced that with the amine to make 4 additional aldehydes. And after these aldehydes were made, we have a total of 10 aldehydes. We actually reduced them using sodium borohydride to get benzyl alcohol, because our chemistry required benzyl alcohol to make compounds of series 3 to 5. So the chemistry to make series 3 and series 4 compound are shown in this box here. Basically, we are doing electrophilic aromatic substitution using the benzyl alcohol on phenol, and that makes the alkylation at number 1 position of the naphthol. And then we react the resulting compound of series 6 with appropriate length of bromo alcohol, and we get compound of series 3 and series 4. And there were no problems. We were able to make these compounds. To make compound of series 5, we started with 6-bromonaphthol, and we first methylated it to protect the hydroxy group, and then we performed Ullmann condensation between the nitrogen and the carbon using copper iodide and phenanthroline. And the reaction worked well. Now on that compound, we performed electrophilic aromatic substitution using benzyl alcohol in presence of BF3 etherate to get compounds of series 5. Now, we hypothesized that in this particular case, the benzyl group is going to attach at the ortho position to nitrogen, because nitrogen, you probably know that in electrophilic aromatic substitution, the most activating group is actually amine, followed by oxygen-containing functional groups. So based on that, we predicted that this product would form, and we did get a product. And when we looked at its, NMR, mass spec, and carbon-13, and they matched with the product. So then we looked at the biological activity of the compound. And if you see series 5 compound, which had a structure similar to that of raloxifene, it showed pretty good activity. Again, the tamoxifen has the activity of 31 micromolar, whereas almost all compound of series 5 had single-digit micromolar activity concentration. Series 2 compounds were comparatively less active. Series 3 compound and series 4 compound also had, single-digit micromolar activity for the IC50. And there was an interesting observation that in all of these series, whenever the substituent was morpholine, the activity was poor. So you see this line right here. In all these cases, the activity is poor. And for some reason, the oxygen in the morpholine, the polar atom there was somehow, affecting the activity poorly. So after having seen the activity, we were quite happy with that we had a compound that showed better activity than tamoxifen. Then we said, okay, let us find out whether our compound binds to estrogen receptor alpha or not. And for that, we used an NR peptide colorimetric ELISA test. So we bought the ELISA kit from a company, and we tested that with the positive control, which was estradiol. So E2 stands for estradiol, and the negative control, which is the tamoxifen TEM. And both of these compounds were actually at the concentration of 25 micromolar. And you can see that if the compound is similar to negative control, then the bar is going to be shorter. And if the compound is similar to positive control or an agonist, then the bar is going to be larger. And for all of the series of compound, I selectively chose compound, the series 3 compound is 3H, and 4H and 5H and 6G. And in all of these compound, whereas we did the concentration of the positive and negative control at 25 micromolar, for my compounds, the tests were done at 25 micromolar and 2.5 micromolar, so 10-fold dilute concentration. And all of these were actually showing that these compounds were working as the negative control like tamoxifen. So they all appear to be showing antagonistic activity. We saw, okay, we got an improvement after we made structure similar to raloxifene. How about we modify the structure even further and make it? So again, so we have the series 3 compound in the middle, and they were showing good activity. On the left-hand side, there is estrone molecule. On the right-hand side, there is this estradiol molecule. So we made analogs similar to estrone by having a ketone group in series 7 compound. Then we made series 8 compound where it has a secondary alcohol, which is similar to the secondary alcohol of estradiol. And then we also made series 9 compound where we had a tertiary alcohol at that position. Further, raloxifene has 2 hydroxy group, whereas my series 5 compound has a methoxy and a hydroxy. So I thought it will be actually better if we have 2 hydroxy, and that will actually resemble pretty similar to raloxifene. So we made series 10 compound, 10 examples, of series 10 compound. So quickly, synthesis, again, synthesis was similar to what we did for series 3 and 4 compound previously. So with series 6 compound, we did the SN2 reaction with chloroacetone, and the product series 7 compound, we got 10 of those compound. And then by simple borohydride reduction, we got series 8 compound. And by, Grignard reaction with the methyl magnesium iodide, we got the series 9 compound. So these chemistry were pretty straightforward. For the series 10 compound, all we needed to do was to actually replace the methoxy with a hydrogen. So basically cleave the ether bond and get series 10 product. We tried different conditions for demethylation and it did not work. Those who are familiar with this type of deprotection, sometimes this methoxy substituent, the deprotection of methoxy ether is actually hard. And one of the easiest group to remove is actually benzyloxy group. So we thought, okay, we may have to go back to the first step and do the synthesis with having a benzyloxy group and then, make series 10 compound that way. So to make series 10 compound, we went back to our, 6-bromo-2-naphthol and we benzylated the hydroxy group. Then we did the Ullmann coupling, followed by the the benzylation using the BF3 etherate, and then hydrogenation, and we were able to remove the benzyloxy group and get series 10 compounds. So after we made all these compounds like series 7, 8, 9, and 10 of each, then we tested the cytotoxicity data. And here are the cytotoxicity data. So basically, the box in green color are my series 7 compounds. The box in black color are my series 8 compound. The box in blue color are my series 9 compounds. And the box in series red are my series 10 compounds. And basically, we are looking at the IC50 value. And since the compound is going to be more potent if it shows the activity at lower concentration. That means smaller the bar, better the activity. And for comparison, we have the data for tamoxifen and raloxifene on the extreme left side, and then rest of the data is there. So as you can notice that we have a number of compounds that are more potent than tamoxifen and raloxifene. However, the series of compound on which I was most hopeful, the series 10 compound, they were actually less active than tamoxifen and raloxifene. So this was a disappointment. However, this disappointment was actually, it rose more suspicion than disappointment. We were, as I told you earlier, we were not sure whether the benzylation would happen ortho to nitrogen or ortho to oxygen. So we decided to actually confirm whether the benzylation happened at the right place or not. But before that, we did some further work with some selected compound. We specially selected the compound which are more active, like with the smaller bars. We selected those compounds, and then we did some more cytotoxicity data with other cell lines. So MCF7 cell line, which is a breast cancer cell line, it is actually high in ER alpha and it has low ER beta. So we wanted to know whether our compound is active on ER alpha or ER beta, or it is non-selective. So to understand the selectivity, we did these tests. The MDA 231B has no ER alpha and it has low ER beta. The LN-CAP cell line is a prostate cancer cell line, which has no ER alpha, it has only ER beta. And MCF10A does not contain, any estrogen receptor. So basically, if my compounds show good activity for MCF7, that means it is actually selectively binding to ER alpha. And when it was tested with the different molecules, and we indeed find the strongest activity in MCF7, as you can see in the bar graph. The structures shown in red are the ones that were chosen for, this test, and none of the series 10 compounds were chosen because they did not show good enough biological activity. So as I said, I was wondering about, whether we got the right product or not. So as I said, we anticipated that the electrophilic aromatic substitution would happen at ortho to nitrogen because amines are more activating. And but if the oxygen, if the benzylation happens right next to oxygen, then the other possible compound is going to be the one shown in left. Now, if you think about the proton NMR of these two compounds, they are going to be virtually identical. Even carbon-13 compound is going to be virtually identical. Also, the mass spec data is also going to be the same because these are isomers. So using NMR, it's actually very hard to tell which one is which. But there are certain types of NMR spectroscopy that can be performed on compounds to actually find out, whether the structure is correct or not. And these are some advanced NMR spectrum. So the other series of compound, when we had the benzylated product, again, if the benzylation happens next to nitrogen and then you do debenzylation, the product on the right is what you will see. And if the benzylation happens next to oxygen and then you do remove the benzyl group, you will see the product at bottom left. So again, it will be very hard to distinguish between these two compounds just based on NMR and mass spectrum. So we performed the noisy and X-ray experiment on these compounds. So noisy is actually a nuclear over Hauser spectroscopy. It's an advanced NMR spectroscopy that helps in identifying where the substitution has happened. And I'm going to briefly explain that. So, we were able to actually prove that the benzylation happened next to oxygen, not next to nitrogen. So the compound that we were initially thinking we had formed was actually not formed. And this explained why the activity was not seen when the benzyl group was removed. So here is the data of the nuclear over Hauser effect. Just to briefly explain to you what happens in NOE spectra is that you irradiate a particular peak. And when you irradiate the peak, that particular peak actually shows inverted in the resulting spectra. And all other peaks do not show, only those peaks show which are actually closer to the irradiated peak. Okay, so the normal NMR spectrum is at the bottom for the molecule shown there. When this CH2 group shown in blue, which is the CH2 attached to aromatic carbon, that CH2 was irradiated. So this particular peak here, it actually became inverted after irradiation. And then protons which are closer to CH2 actually get enhanced and all other protons become invisible. So as you can see, these two peaks here, which are actually got enhanced, these are peaks for these two CHs of the benzene and that one. In another experiment, this red CH2 was irradiated, which is an ether CH2. Okay, so it shows up at about 5.2 ppm or so. And after irradiation, the peak became inverted, and these protons showed up as the enhanced peak, and the rest were invisible. If the CH2 had attached next to nitrogen, then we would have seen some enhancement at the NCH2s, which we did not see at all. So NCH2s are in this area, and we did not see any enhancement in this area. So this clearly showed that the substitution happened next to oxygen, not next to nitrogen. So this
Could you
was,
please conclude? I'm so sorry to interrupt, but we have another session in line. So,
okay.
Within 5-7 minutes, if you could conclude.
Sure, I will try. Yes, I'm sure I should be able to do that.
Okay.
So The X-ray structure further proved it. And as you can see, the X-ray structure, the benzyl group were at the ortho position to oxygen. We did some more cytotoxicity data from the National Cancer Institute in US, and they basically were consistent with the preliminary results that we had obtained. And they selected 10 of my compounds and did the activity on them. So they initially did 10 micromolar concentration on the 60 cell lines, and then they did 5-dose cytotoxicity against the same set of cell lines for the selected compounds, and they were found to be pretty active. We performed the in vitro pharmacokinetics to understand whether this compound is going to be stable under the in vivo conditions or in vitro pharmacokinetic conditions. So we performed what we called a membrane permeability data using the PAMPA assay, and the compound were found to permeate through the cell membrane or PAMPA membrane actually in a reasonable rate. Metabolic stability as compared to the drug propranolol, the compounds were actually more stable than propranolol in human liver microsomes, and the compounds actually were, even after 1 hour of incubation, the compounds, a significant amount of compound was actually left. So they were not getting metabolized right away after going inside the body. Looking at the toxicity, the compounds were actually tested at 300 milligram per kg body weight of live rats, and none of the rats died during the experiment. So they were found to be pretty safe to ingest to the animals. I'm just going to skip the more design part because, just to save some time. Now in the new design, we are actually trying to make compound having the steroid structure with the side chain. And for that, we have developed some chemistry, which I'm just going to very quickly show you, that we are designing the steroidal structure, and the AR group will have the side chain. And then we are going to test these compounds because they have similar structure to estradiol. And we have developed this chemistry already. Another steroidal structure that we have developed is shown here, where we have been able to make these steroidal compounds, and we are in the process of attaching the right side chain at the R position. The chemistry is shown in this scheme here. Another scheme that is leading to a similar steroidal structure is also underway, and we have made several examples of this already. And in conclusion, we have developed several series of molecules which are actually binding to the estrogen receptor alpha, and at the same time, they're showing significant cytotoxicity. And these compounds appear to be antagonistic to estrogen receptor alpha. In future, we plan to make more compound and fine-tune the ER binding activity as well as the cytotoxicity studies. And we will perform in vivo pharmacokinetics and in vivo efficacy. And in fact, I'm looking for collaborators who could do the in vivo work for our compounds. And finally, I want to thank my group. I had a number of, graduate, undergraduate, postdocs work in my lab. I had several visiting PhD students from India. Their names are actually with the asterisk. You can see their names are listed there. And the funding agencies that made this work possible. And finally, I would like to thank you guys for listening. Thank you.
Thank you, Professor Jha, for a very informative session. It is indeed a moment of pride and joy when fellow Indians excel in their respective fields abroad. They bring a good name to the country. And certainly, it would be a pleasure to host you in Deshbandhu College in person whenever you are in India. And on behalf of Department of Chemistry, Deshbandhu College, please accept e-certificate and e-memento as a token of appreciation.
Professor Amitabh Jha, sir, please stop your sharing.
Sure. Thank you. Am I able to stop it? Is it stopped yet?
No, not yet.
So, so you, okay, stop share. Okay.
Right. It's fine now. Ashwini, please e-memento and e-certificate to the esteemed speaker. Ashwini.
Yes, ma'am. It's sharing. It's sharing.
Not showing. It's not showing. Participants, we may not be able to take up the questions due to time constraint. We'll share the email ID of the speaker. You can get in touch through email. And
Thank
it is,
you.
I know it is a little inconvenient time. It's early morning, 3 a.m. in Canada. So our sincere apologies to the speaker. And thank you once again. Thank you for accepting the invitation. Thank you, Professor Jha. Okay.
Thank you.
Thank you once again.
Bye-bye.
Over to Parul. Over to Parul for next session.
Thank you, Dr. Aparna. I would like to extend my sincere thanks to both of the speakers, Dr. Partha Sarthi Mukherjee and Professor Amitabh Jha, for a very interesting talk. And thanks is due to our chairpersons, Dr. Aditya Saxena and Dr. Aparna Sekhar. And thanks to all the participants for making this event successful. And I request Dr. Aswani to present a token of appreciation to the chairpersons, Dr. Aditya Saxena and Dr. Aparna Sekhar.
मैम शेयर हो रहा है? Is it share?
No, it's not.
We can't see anything, Ashwini.
मैम actually मैं यहाँ से visible है.
Yes, it's visible. It's visible.
Now it's visible.
Okay, ma'am.
The feedback form would be shared in the chat box for this session. And over to Parul for next session.
Yeah. And due to time constraint, we don't have any tea break and we are starting the next session. So welcome you all again in the section 4 of International E-Conference on Recent Advancement in Chemical Sciences. Dr. Ashwini, please mute yourself. Welcome you all again in the session 4 of International E-Conference on Recent Advancement in Chemical Sciences. I welcome our next speakers of the session, Professor Niranjan Parajuli from Central Department of Chemistry, Tribhuvan University, Kathmandu, Nepal. And the next speaker is Dr. Rais Ahmed Khan from Department of Chemistry, King Saud's University, Kingdom of Saudi Arabia. Welcome both the invited speakers. And it is a matter of pride for us. We have two chairpersons with us, Dr. Manisha Sanger, Associate Professor in Department of Geology, Deshbandhu College. She herself is a good researcher and a very active faculty member of the Deshbandhu College. We have with us Dr. Indrakant Kumar Singh, Associate Professor of Geology, Deshbandhu College. He is coordinator of DBT Star College Scheme, Deshbandhu College. Dr. Singh is a determined researcher with many publications and award in his credit. And he is really a motivation for many of us in the college. Welcome, sir. May I now invite Dr. Manisha Sanger, ma'am, to conduct the session further. Over to you, ma'am.
Thank you, Parul. Thank you so much. Am I audible?
Yes, ma'am, you are audible.
A very good morning to all who have graced this occasion by their auspicious presence. I feel very honored and proud to introduce our speaker, Professor Niranjan Parajuli, to you all. Professor Niranjan is an eminent personality from Central Department of Chemistry, Tribhuvan University, Kathmandu, Nepal. Sir has held many important positions in his tenure of teaching and research, Sir is head department of Sir has been head department of biotechnology in National College Tribhuvan University. He worked as a postdoctoral associate at Department of Chemistry, North Carolina State University, USA. Sir was a postdoctoral associate in the Department of Chemistry, South Korea. Sir has also served as a faculty in Department of Biochemistry, National College Tribhuvan University, and as an assistant professor in the Department of Natural Sciences, Kathmandu University. Professor Niranjan has always been a very meritorious student. He was awarded gold medal and distinction in his post-graduation that paved his career for a research in South Korea. Professor Prajuli has research grants and fundings of over 16 million Nepalese rupees from government-funded agencies like University Grants Commission, Nepal. Sir is a principal investigator for various projects projects such as genetic characterization of actinomycetes useful for antibiotic production. He is Chief Scientific Advisor on the project Investigation on Secondary Metabolites from Natural Products through computational chemistry and immunochemical approaches. Professor Prajuli has been granted a project on screening of extracts from medicinal plants from inhibition of enzymes of diabetic target from a very prestigious agency, the World Academy of Sciences, Italy. Professor Niranjan is a PI for many projects from Nepal Academy and Academy of Science and Technology. Sir has been honored with many prestigious awards as Science and Technology Youth Award by Nepal, Academic of Science and Technology in 2017. Sir received his Mahendra Vidyabhushan Class A award granted and a gold medal granted by His Highness King Gyanendra in 2015 and King Birendra in 2009. Professor Prajuli holds a very important position and many other positions in a number of various research committees. Sir has more than 50 publications to his credit in international and national journals. Sir has Sir has delivered innumerable invited talks as a keynote speaker. Today, Sir will speak on genetic characterization of actinomycetes useful for antibiotic production. We are all enthusiastic to listen to you, sir. The forum is all yours. Welcome again, sir. Thank you for your presence today. Thank you, sir. Over to you, Parul.
Over to you, sir, for delivering your
Over to you, sir.
Okay, thank you very much, Manisha madam, for nice introduction with about me. Now I want to share my file.
Yes, sir.
So is it okay?
Yes, sir.
This file is okay. Thank you very much for nice introduction, and I would like to say good morning to all participant and organizing committee. And I am very happy to present my research with you. And my topic of my today's talk is genetic characterization of actinomycetes useful for antibiotics production. And as we know, antibiotics are very useful, very useful medicine for us. And about 80% of commercially available antibiotics are produced by soil microbes. So my research is focused on isolation of actinomycetes from soil because there is a scarcity of antibiotics in recent days because of emergence of antimicrobial resistance, which has become a global problem and which can be addressed only through the discovery of new or hybrid antibiotics. And so far, around 500 Stoichiometric species are reported, but most of them are uncultured.
I think there is some technical glitch. Niranjan sir, you are not audible, sir. Professor Niranjan is not audible. I think there is some technical glitch from inside. Dr. Umesh, you may contact with him. Am I audible, Dr. Aparna?
Yes, Parul, you are audible, but perhaps there is some glitch at speaker's end.
Yeah.
Even the screen sharing has gone off.
Yes.
Parul, can you hear me?
Yes, I can.
Acha, so I think there is some glitch at speaker's end only.
Yes, I think so. Dr. Umesh, are you there?
एक मिनट में उसको कर, let me call Umesh.
I think we can tell sir to connect again.
हाँ, but मनीषा शायद वो लीव हो गया है उनका. Even in list of participants, it's not showing.
Oh.
Yes.
तो एक बार हमें इन्हें कॉल कर सकते हैं क्या?
मैं उमेश से कोऑर्डिनेट करती हूँ जरा.
या व्हाट्सएप कॉल हम करें एक बार प्लीज देख लो.
हाँ, अगर है, if you have his number, you can please make a WhatsApp call.
अच्छा अच्छा, I'll try. Okay, I'll do that.
I am trying to contact Umesh.
Okay, fine. I'll do that. Okay. Just a minute, huh? I might leave in the process. I'll join again.
No problem.
Okay, ma'am, no problem. Yes.
Manisha, he has joined.
rejoined.
Sir has
Yes.
joined.
Sir, please unmute
Yes,
yourself.
Yes, sir, please. Yes, sir. Please continue. Thank you, sir. Thank you.
Sir, please unmute yourself.
Sir, please unmute yourself. Ashwini, please
Hello.
unmute, sir.
Hello, I'm extremely sorry because I got interrupted. My internet connection, my internet disconnected. I'm very extremely sorry for that. Please.
No problem, sir. It's okay.
Okay.
Sir, it happens, you know, technology has its own limitations. Yes, sir, please start sharing.
Yes,
your-
sir.
Now
Yes.
I will again share my file.
Okay, sir. Thank you.
I think you have to make me co-host for sharing to allow to share my file.
Dr. Ashwani, please do the needful.
Ashwini, are you there? Ashwini has also left, it seems. Let me call him then. Manisha? Manisha, can you hear me?
Yes, Aparna.
Manisha, Aditya host ho gaye. I don't know what, there is some issue.
I'll do something about it. I'll do. Yes.
Sorry for the interruption, sir.
Yeah, no problem.
So technology has its own limitations.
Yes, right.
For last 2 years, we have been using these platforms, and sometimes there are glitches. It is requested to all participants to wait for all sessions to be over, and then you can submit the feedback form. And we are really sorry for the interruption and delay. Kindly be patient and please bear with us.
Okay, I told Monica she is making Ashwini, Dr. Ashwini host again. She's just doing it.
ठीक है, ठीक है. Ashwini is the host now. Ashwini, please make sir co-host. Yes, sir, you are co-host now, sir.
Okay.
Yes, sir.
Please proceed.
Yeah, thank you. Thank you very much. Now I resume my talk. Now, particularly, I'm focusing on isolation of soil microbes, that is actinomycetes. And as we know, the major source of antibiotics are actinomycetes because 80% of commercially available antibiotics are isolated from Stoichiometric. Okay, and you know this antibiotics resistance is a global issue and very emerging emerging issue in health sector. First penicillin antibiotics were discovered in 1928, around 1928, and later on, after a few years, in 1940, some bacteria were seen that show the resistance with the penicillin. Similarly, in 1948, another type of microlide types of antibiotics were discovered, and in 1953, some bacteria also show the resistance with microlide antibiotics also. So there is an urgent need of discovery of new types of antibiotics that can tackle the global emerging problem against multi-drug resistance bacteria. So in my research, I particularly focus on isolation of actinomycetes, particularly from the high altitude in Nepal. And as you know, these actinomycetes are the filamentous gram-positive bacteria, and we can isolate them from soil, water, and also from the marine ecology also. But in Nepal, we don't have marine ecology, so particularly were concentrate isolating those actinomycetes from soil, basically in Nepal. And you know, the actinomycetes show some bacterial character and fungal character as well. As I already discussed, they are the major source of secondary metabolites, including antibiotics. And now I want to share you some structure of antibiotics. These are the polyketide types of antibiotics isolated from Stoichiometric, various types of Stoichiometric species, and so far more than 500 Stoichiometric species were isolated globally in order to isolate the useful secondary metabolites, including antibiotics. And these are some glycopeptide types of antibiotics. And now, main problem in antibiotics research is that some actinomycetes or some Stoichiometric species are unculturable. Now, while talking on these the 2PK506 producer, one from Stoichiometric species, this is Yame 6858 and another Yame 6548. But one of them, this Yame 6858 is unculturable and Yame 6548 is culturable. Similar, so in antibiotics research, particularly in actinomycetes research, most of the actinomycetes are unculturable, even if we identified that they could produce some antibiotics. But if we couldn't culture them in laboratory setting, then we have to find some another alternative like for such as heterologous expression and like that. So in my talk today, I'm not talking on heterologous expression, but I want to share you some ideas. What are the problem lie in antibiotics research? Now it shows some aminoglycosides type of antibiotics like gentamicin, tobramycin, neomycin, all they are aminoglycosides type of antibiotics. And this is the gentamicin biosynthetic gene cluster. We can isolate this type of gene cluster from Micromonospora purpurea or Inospora. And there are several genes involved. And similarly, this is the biosynthetic route for gentamicin, tobramycin, kanamycin, different type of aminoglycosides type of antibiotics. So it is very useful to understand their biosynthetic pathway in bacteria in order to discover the hybrid or new type of antibiotics. And this is the another type of antibiotics, this doxorubicin, and this is anthracycline type of antibiotics. And it is also shows the anti-tumor activity also. And this type of antibiotics, doxorubicin, is also isolated from Stoichiometric species, Stoichiometric persistens. And this is another type of antibiotics, and it shows this slide shows the biosynthetic gene cluster of erythromycin. And so all those antibiotics were isolated from soil microorganisms, soil microbes, Stoichiometric species, and it shows the overview how we can clone biosynthetic gene cluster, biosynthetic gene cluster based on actinomycetes. First of all, we isolate the genomic DNA, and later on we prepare the cosmate library, and we perform the colony hybridization, selecting the particular biosynthetic gene cluster, and after performing sequencing, then we come to know the biosynthetic pathway of any secondary metabolites or antibiotics. Now I want to move on my particular focus area, experimental procedure, what I did in Nepal. Now, as I already mentioned, my main target is to isolate Stoichiometric species in order to isolate, or in order to annotate the antibiotics or secondary metabolites. So I collected my group collected some soil samples from high altitude. Then we use ISP4 media for to isolate Stoichiometric species, and after that we perform the flax fermentation in TSP media. And after that, we isolated metabolites using ethyl acetate solvent, and we perform the antimicrobial test and determine the MIC and MBC also. And finally, we send out sample for liquid chromatography, high resolution mass spectrometry. And similarly, on the other side, for molecular characterization, we amplified their 16 years RNA by PCR, and then we perform the sequencing to identify their taxonomy, and we identified particular Stoichiometric species. So regarding isolation of actinomycetes, my group performed experiment as shown in this slide. Is first of all, we took 1 gram of soil and put it into 9 ml distilled water. Then we perform serial dilution as shown in this slide, and then we use ISP4 plate, ISP4 media, solid culture media in order to isolate Stoichiometric or actinomycetes. And you can use the another plate also, ISP2 plate also, and there are several other protocols also for isolating actinomycetes, but in our best knowledge, we optimize our protocol on ISP4 culture media. So in my research, particularly we are using 2 solid culture, ISP4 and another TSP for fermentation, as a fermentation media. Instead of using these 2 media, you can use other type of media also for isolating actinomycetes. Let me organize this. This cursor is not helping me. Okay, and after that, we isolated the secondary metabolites from culture broth using TSP media, and sometimes we also use the wheat bran media, and the ethyl acetate was used for extracting secondary metabolites. And we also perform the antimicrobial test by agar oil diffusion method to know their antimicrobial efficacy. And similarly, we also perform the MIC and MBC by resazurin-based assay. And then finally, we analyze in bacterial extract through LCMS and LCHRMS. And for this facility, we didn't have this facility in Nepal. So We send our sample to Lucknow Sophisticated Analytical Instrument Facility in India, and sometimes we also send our sample to South Korea and Germany for the same process, for the same analysis. And later on, we work on metabolomics, taking the raw data of mass spectrometry, and basically my group concentrate on these software for molecular annotation, Mastronoma, GNPS, Sirius, and some other software also we use. In coming slide, I will show you this. And in order to identify their taxonomy, we isolated their genomic DNA using phenylchloroform method. This is the very standard protocol described elsewhere. And similarly, we amplified their 16 years rRNA for taxonomy identification using PCR using the standard set of primer. And finally, we determine the sequence of this amplified 16 years RNA by sequencing. And we use this protocol. We use Taq polymerase for PCR amplification. We use PCR premix of as shown here, and we use this forward primer and universal forward primer and reverse primer for identification of for identification or amplification of 16 years rRNA of bacteria. And this is our PCR condition for amplification of the same open reading frame 16 years rRNA. And yeah, in this research, we have isolated actually more than 50 different actinomycetes from the soil collected from diverse locations of Nepal. And as you know, these actinomycetes are filamentous bacteria, and they have a peculiar growth pattern. And they have a different color also. Some actinomycetes are dark gray, and some are just light gray, dark brown, and white yellowish, like that. And we identified isolated actinomycetes through morphology, through their morphology, and through gram staining also. We also perform the gram staining because actinomycetes or Stoichiometric species are gram-positive bacteria. So we also perform this gram staining also for identification of their morphology. And later on, we perform the molecular sequencing, as I already mentioned in previous slide. Also, and it shows the primary screening of those actinomycetes, whether they could inhibit the microbial growth or not. And we use Klebsiella pneumoniae, Shigella sonnei, E. coli, Salmonella typhi, and Staphylococcus aureus. And this slide shows the perpendicular screening of bacterial extract. And it shows the antimicrobial assay of bacterial extract, and we have several number of isolates, but here I want to talk particularly on few of them, like BN14, sample BN14, and it shows this slide shows the very good inhibitory effect of this BN14 as compared with the positive control. Here, as a positive control, we have used neomycin of concentration 1 mg per ml, and we found this bacterial extract, BN14 isolate, shows the better zone of inhibition as compared to positive control neomycin. And even if we know this BN14 is not a pure compound, we have to purify this. Even if in a crude extract shows the better zone of inhibition than neomycin positive control. And it shows the antimicrobial assays, this zone of inhibition with bacterial extract of various sample, and this BN14 shows a very promising antimicrobial effect with various bacteria. And here in this experiment, we also perform the liquid fermentation and solid fermentation both. For liquid fermentation, in this research, we did not use the bioreactor for fermentation of Acknowledgements. Here we use the flax. Simply, we use culture flax or flax fermentation, widely known as flax fermentation, in 50 ml volume, or sometimes we use 100 ml volume, media volume also for flax fermentation. And yeah, you can see your asterisk sign. This BN4 asterisk. This asterisk meaning this is we also perform solid state fermentation in some cases, and this we found the solid state fermentation and liquid state fermentation. So a little bit the same pattern of antimicrobial effect. And we also determine the MIC and MBC of BN14 extract, and this slide also confirm that this BN14 shows the better inhibitory effect as compared to neomycin. And for molecular identification of isolates, we isolated genomic DNA of isolates or actinomycetes, and this is the 10 kb ladder, and we use 0.5% agar gel to run this genomic DNA on electrophoresis. And this is the PCR amplification of 16 years rRNA, and we use the universal primers 27F and 1492R for amplification of these 16 years rRNA. And before sequencing, we purified them. We purified this PCR product using standard kit protocol, as described by supplier or vendor. And then, as I already mentioned, we perform the solid fermentation and liquid fermentation using TSP media, tryptic soy broth, and using ethyl acetate. We extracted metabolites from bacterial extract, and then we analyze that ethyl acetate fraction using liquid chromatography mass spectrometry. In some cases, we use low resolution LCMS, and in some cases, we use high resolution mass spectrometry for analysis. And analyzed by this sample. In this particular sample, BN14, we analyze Mastronova 12 software and compared to database library and literature and dictionary of natural product and Matlin metabolite searching cloud. And we found those metabolites are present in sample BN14. And here, in this research for molecular annotation, we use this Mastronova. We use Mastronova just to open the raw data of mass spectrometry. And for database searching and for molecular annotation, we use Sirius database, and for molecular networking, we use GNPS software also. And this is the just raw data analysis in Mastronova, and after identifying some parents peak, we compared those peaks. We sourced those peaks in various databases, as mentioned here, like Matlin and Sirius and anti-waste database also. We use anti-waste database also, and natural product Atlas also. And then we annotated some useful molecules also. And some molecules are mentioned here. These through molecular annotation through metabolomics. And yeah, GNPS approach suggested the crude extract of these BN6 and BN14 may have those those molecules, and the series alone with the additional chemical scaffold like bacterial alkaloids also reported, and enthromycin type of antibiotics are also reported. And now these are the some antibiotics annotated through GNPS, server and series platform in sample BN14. So now I come to conclusion. The production of secondary metabolites from actinomycetes differ according to constituent and type of media used, and more biological active actinomycetes strain are prevalent in Nepalese soil than commercial antibiotics producing actinomycetes, and the isolated actinomycetes might be significant to cope with the current challenges of emerging MDR pathogens. Thus, bioprospecting natural products from actinomycetes of Nepalese origin could be potential source for discovery of novel antibiotics and anti-tumor agent in future. I would like to thank University Grant Commission Nepal for financial support. I would like to thank Sophisticated Analytical Instrument Facility Lucknow for LCHRMS raw data, and I will also thank this Microgen South Korea for DNA sequencing, and these two students are also actively participated in this research, Sovika Bandari and Srishti Sureshta, and these are the references, and this is my university, Tiruvana University. It is the oldest and a public university, and in terms of enrollment of student, it is the 10th largest university in the world, and its rank is among 800 to 1,000 best universities of the world by Times Higher Education ranking. Okay, thank you very much. So please, over to you. If you have any queries, let me know.
Thank you very much, sir, for very informative talk. If anyone has any questions, 1 or 2 questions, please post in the chat box. Thank you very much, sir.
Yeah, welcome.
And kindly accept our token of appreciation from
Please,
Deshbandhu
sir.
College.
Please, sir, stop your sharing, sir.
Just a moment. Or stop sharing. Okay.
Dr. Ashwani, please present token of appreciation. Please accept, sir, from our side.
Yeah, thank you very much. Thank you very much for giving me this opportunity. And I am very thankful to Dr. Umesh for inviting me in this platform. And this is my first opportunity to discuss my research with you all, very brilliant researcher in India. And I'm looking for, at this moment, I also want to say something. I'm also looking for collaborator in metabolomics study. So if you want to collaborate with me in terms of metabolomics molecular annotation, please kindly email. Thank you very much.
Sure, sir. Any of the participant can collaborate with sir if they wish.
Thank you, Dr. Niranjan. I will very soon. I'm going to approach you.
Yeah, thank you, sir.
Thank you.
Yeah.
So do you have all the system for analysis as well, or trained person with you?
Yeah,
Because
yeah,
I have a
yeah.
lot of data lying with me, and there we need a kind of limitations is to analysis. Yeah.
Yeah, in my case, our main problem is LCMS.
Yeah.
For data analysis, my group can manage all
Great.
these things.
Great. Thank you so much.
Yeah, welcome, sir.
Thank you, sir.
Yeah, welcome. Welcome, ma'am.
Now I invite Dr. Indrakant Kesing for conduct next session.
Thank you, Dr. Parul, for this opportunity. And so the next speaker of this session is Dr. Raiz Ahmad Khan. And before I will request Professor Khan to take over the session forward. So first, let me introduce formally that Dr. Raiz Ahmad Khan has received his PhD in chemistry in 2012 from Aligarh Muslim University, India, under the guidance of Professor Sartaj Tabassum. During his PhD, he was awarded research fellowship in Indo-Italian exchange program with Professor Claudio Paterni and Professor Fabio Marchetti, University of Cameroon, Italy. In 2012, he has accepted the assistant professorship in chemistry at King Saudi University, and he will work with Professor Jan Reddick, University of Leiden, in Netherlands, for the period of 4 years. In 2019, he has been promoted to associate professor in chemistry at King Saudi University, and he has published more than 85 research articles of international repute, and he has won US patents to his credit. His area of research focuses on development of metal-based anti-cancer agents and their interactions with biomolecules, antiquorum sensing and inhibitors, and anti-biofilm agents, environmental science, etc. So before wait further, we are very excited to listen Professor Raiz Ahmad Khan, because Dr. Umesh and even myself have a lot of interest and very excited to listen, specifically work related to metal-based anti-cancer agents. So we are eagerly waiting for you, sir. Yeah, thank you. Please, you can. We are happy to deliver. Receive your. Yeah. Anything else, Dr. Parul? Dr. Raiz Khan is, I think, I hope.
Thank you so much for inviting me for this talk, and thank you so much for I appreciate the kind words that you have spoken about me, Dr. Indrakant Kumar Singh. Yeah, I'm really happy to be part of this conference and wish you a very best. And I hope now the conference has been almost concluded with the last talk. I guess so. I'm just trying to share my screen. Is my screen is okay and I'm audible?
Yes, sir. You are audible, sir.
Yeah. Can you see my screen?
Yeah, Dr. Khan, you can just full screen is completely. Yeah.
Okay. Yeah. As I have given my topic as unveiling the human serum albumin binding affinity and anti-cancer potential of some of the bioactive organic moieties appended with organometallic ruthenium compounds, especially with paracymene. So, yeah, what's happened? Yeah. As we know that the metal drugs have been employed over centuries to palliate different kinds of fatal diseases, especially cancer. However, the boundary line between toxic and therapeutic doses caused the scientists to deeply research drug discovery. As a consequence, the door of the modern era of research on metal drugs was opened when Michael Perron discovered splatin in 1844, and its anti-cancer activity came into limelight in 1965 by Rosenberg, that he reported the efficacy of the treatments of various cancers such as lung, head, neck, esophageal, testicular, and ovarian cancer. Alongside cisplatin, many other platinum-based drugs such as carboplatin, oxaliplatin, nedaplatin were found to be potent anti-cancer agents. These complexes were efficacious against a majority of cancers, but several drawbacks such as poor solubility, low selectivity, drug resistance, and high level of in vivo toxicity, particularly neurotoxicity, nephrotoxicity, thrombocytopenia, and myelosuppression, neutropenia, and neuropathy have limited their uses. The challenge ahead was there to develop new and more effective metal-based anti-cancer agents. On that basis, the scientists has developed new things. They have started their race over search of new metal. In that research, the ruthenium complexes appeared as unarguable among the most important potential candidates investigated as anti-cancer metal drugs other than platinum. Their inherent properties like mimicking iron, redox, accessible oxidation state, low toxicity, smooth interaction, and binding with DNA, protein, and other biomolecules. The molecules that fascinated the interest of the research community was with few of the ruthenium compounds that have come up as an that has gone to a first clinical trial, and that were NAMI. NAMI is just an ruthenium compound with the imidazole in it, and outside also, as you can see there, it is like with ruthenium 4 tetrachloride and 1 DMSO group. Then there is imidazole and this another as positive imidazole. Okay, then there is another class that came as known as KP1019. The molecules were quite similar. It was with an indazole moiety. It had 2 indazole groups with 4 chloride and 1 indazole group outside it. The third compound that fascinated the people were NKP1339. It is just a sodium salt of KP1019. Just to know and have an interest to go further in detail, I would like to share some details about one of the compound that leads the ruthenium compound molecules, ruthenium metal-based drugs. This is like the drug NAMI, NAMI-A. Okay, NAMI-A is a ruthenium-based anti-cancer drug and is one of the 2 ruthenium-based drugs that have been entered clinical trial. NAMI is considered a prodrug formulation that becomes active upon hydrolysis. That means it is not active, but when it gets hydrolyzed, it becomes active. So at pH 7.4, a chloride is exchanged with a water, as you can see here, a molecule which neutralizes the molecule's -1 charge, and upon lower pH, imidazole cleaved and replaced with water. At the pH goes when 3 to 5, the imidazole gets out of it. Unlike cisplatin, a platinum-based drug, it has been shown in vivo that NAMI-A doesn't inhibit primary tumor growth, but decreases the speed at which metastasis occurs. So that's the different mechanism that this drug has, or the drug candidate has put in, is that it is not killing any cell. However, it is just decreasing the speed of the cell. metastasis. It has also been found that when compared to highly cytotoxic platinum compounds, NAMI-A is much less cytotoxic and has a differing mechanism of action. As I have already told that in vivo reduction of ruthenium products into ruthenium-2 complexes can be better explained by the hypothesis of just a second. Yeah, hypothesis of and usually tumor cells have a low oxygen content, that is we call as hypoxia, and lower pH due to the excess production of lactic acid by glycolysis. Related to the surrounding normal cells, therefore a relatively high production of ruthenium-2 in tumor cells is favored. Ruthenium-2 complexes are more reactive and can coordinate biomolecules more firmly to provide selective toxicity. Among the ruthenium complexes, the area that fascinated recently, I should not say recently, but it is in 1992 when it was emerged. It is half sandwich arene ruthenium complexes. It offers great promises for exploration as anti-cancer agent. This is because of the vast structural diversity and bonding modes. For example, the hydrophobic arene ligand facilitates diffusion of the drugs through the cell membrane, whereas the three remaining coordination sites, usually occupied by various mono, bi, or tridentated ligands, offer a diverse structural and coordination mode and modulate biological properties of these complexes. The first molecule that catches the attention of the researchers was given by Tocher in 1992, and it was with the benzene as an arene and two dichloro groups, and the drug that they have attached is metronidazole, that is an anti-cancer drug. Okay, so in this field, the extensive investigation of ruthenium arenes in respect of anti-cancer activity is pioneered by Sadler and Dyson. These two groups works extremely with this class of compounds. The what you call as the primary or the lead compound that this group has given is RM175, that is a biphenyl ruthenium ethylene diamine chloride compound with the PF6 as ion. It was reported by Sadler. Secondly, is the Rapta-C. This Rapta-C is just ruthenium parathymine as an arene, and PTA, that is 1,3,5-triaza 7-phosphotricyclo-3311 decane as this group, developed by Dyson. They have shown a promising therapeutic potential and are considered to be the prototypes for anti-cancer arene ruthenium complexes for the design and understanding of the mode of action of other anti-cancer drugs. Despite the investigation in this direction, development and improvement in the selectivity and activity of such complexes is of prime importance to develop safe and selective and more effective arene ruthenium-based biological agents. Yeah, okay. Apart from supposedly low general toxicity and a supposed highly selective ruthenium compounds for cancer cells, the main reason for flourishing design of ruthenium arene-based anti-cancer drugs are the amphiphilic properties of the arene ruthenium unit, provided by the hydrophobic arene ligand, counterbalanced by hydrophilic metal center, and the synthetic diversity of arene ligand, which is an excellent scaffold for the coupling of organic segments for targeted chemotherapy. Another important feature is the hydrolysis of ruthenium halogen bonds to give ruthenium aqua species, while the arene ruthenium bond is robust. The corresponding aqua complexes will exist over a range of pH that is around 7.4 and 7. But for pH that is less than pK, is greater than pK, the hydroxo complex formed by deprotonation will be predominant. As hydroxides is a less labile ligand than water, it will not so easily be displaced by biomolecule targets. Equation of chlorocomplexes might be suppressed extracellularly by high chloride concentration, that is 0.1 molar, but becomes possible after the complex enters the cell due to low chloride concentration, that is 4 to 25 millimole found intracellularly. The leading example, Raptor is in the preclinical trial because of its remarkable anti-metastatic, anti-angiogenic, and anti-tumor activities. However, the in vivo anti-tumor activity of kinetically and thermodynamically unstable ruthenium-3 complexes was discovered when reduced to stable ruthenium-2 complexes under hypoxia, pH 6.7, and at the level of high glutathione. So with this brief introduction, I think it could be a big one for other people. I would like to come up what we are doing in this. We are now designing new organic motifs that are biologically compatible, that are means more towards have some biological application. And corresponding to that, we are synthesizing parathymine ruthenium compounds. We used to characterize them using various spectroscopic techniques like UV, FTIR, proton, C13 NMR, SIMS, and of course at the end, single X-ray crystallography. We support our the characterization also with theoretical chemistry using computational chemistry. In earlier, one of my paper came up with Dr. Rita Kakkar and Dr. Deepti Gupta that I had done in my PhD, and that appeared in organometallic chemistry. In that we have discussed about the bond formed between ruthenium, chlorine, and the tin. Means the bridging chlorine between ruthenium and tin that we had reported for the first time, and by the help of Dr. Deepti Gupta and Dr. Rita Kakkar from DU, and I think I know that Dr. Deepti is the in the organizing committee also, so she helped me in that because we met before. And the next step that we do is we used to interact these molecules with biomolecules. Means these compounds with biomolecules like protein, like HSA, BSA, DNA, etc., using various spectroscopic and fluorescence, means spectroscopic techniques like UV, fluorescence, circular dichroism that is used for chiral compounds. After the interaction with the biomolecules, we go for in vitro cytotoxicity of these compounds against various human cell lines with different histopathological origins. Histopathological origins means with different origin in the body, like neural or lung, breast, testicular, oral, liver, etc. Means with different body parts we used to evaluate them. Earlier in my PhD, I was helped by ACTRAC, that is the Tata Memorial Cancer Institute, by I think Professor Surekha, if I am remembering exactly the name. Then after that, we see the migration of these molecules via cell imaging. Means the important part is that how you can say that your molecule is entering somewhere, how the molecule is entering in a cell, does it reaches to the cell or not? Okay, that's a big concern, and that we tried with different staining with different dyes with cell imaging. Okay, after that we go for a cell cycle analysis, in that we study at which phase our compound is still stopping the cell division or the formation of the new cell. That is the main concern with the cancer and cancer cells. After that, we used to see the mechanism of how it is working, either with apoptosis or necrosis, or what is these molecules are doing. And when we have good opportunity and we have some molecules that are good in up to this level, then we go for sometimes gene expressions. Means we choose few genes that are responsible for the cancer, and we used to study by using RT-PCR with this. RT-PCR means real-time PCR. Okay. After that, we used to go for in vivo toxicity profiling for these compounds, because the toxicity is a very, very important concern that need to be addressed with a metal complex. Okay, these are the some of the molecules that we have just published with some of our bachelor students. Okay, this is we have published one with ruthenium, and there is a with this naphthaldehyde and this thiazole moiety. That thiazole moiety is quite active, and they are present in many of the pharmaceutical drugs. So we have just taken them and just made a simple shift base just to make the audience understandable. I have taken very, very basic examples that we are doing. We have synthesized. Okay. Similarly, we have published an inorganic chemical actor recently, and that is a also with the thiazole, but thiazole with the same with the different aldehydes. And here if we didn't use nitrogen, so there is a rearrangement in this, and we got a in all products. And then there is another one that is very, very basic. Here we have taken just amino benzimidazole, because this compound has a property of crossing the blood-brain barrier membrane of the cell. Okay, so just we had taken with this, and we have just substituted one with methyl group, and we have made this complex with ruthenium. We obtain with characterize these compounds with numerous characterization techniques like IR, NMR, ESIMS, UV, etc. And finally, we got the single X-ray structures of them also. So these are the three molecules that we are showing here. Okay, so now we can after this synthesizing these complexes, we had done the binding studies of this with human serum albumin, as my topic is more concerned about HSA. Okay, the major plasma protein, human serum albumin, acts as a depot and carrier for many exogenous molecules and endogenous molecules. So the exogenous like drugs, nutrients, etc., and endogenous like fatty acids, bilirubin, etc., in the blood, and plays a vital purposes. The binding affinity of the drug or potential drug molecules to HSA controls the active free concentration of the drug, and thus substantially affect the pharmacodynamics and pharmacokinetic profile. Therefore, the interaction study of potential drug molecules with HSA is significant, as it can help understand the drug deposition and elimination, as well as drug delivery and efficacy. So we started our experiment with a very basic one. We have studied the intrinsic fluorescence, okay, using the fluorospectrophotometer, sorry. In studying the interaction of small molecules such as drug with protein, steady state fluorescence is one of the most routinely used technique. This tool is simple and less time consuming, and at the same time, it provides numerous useful parameters related to binding. In the fluorescence quenching, the alteration in the native fluorescence emission signal of the proteins are recorded, and the data is analyzed using a set of standard equations. The fluorescence emission of HSA in the absence and presence of the compounds are presented in the figure. Okay, as you can see, it is moving from the peak maximum roughly at around 340, and the concentration of the compounds was increased from 0 to 3.3 micromolar. Okay, Moreover, a prominent blue shift was observed when the compounds were titrated to HSA. The fluorescence quenching and the blue shift clearly shows the formation of protein ligand complex between HSA and the compounds. The primary analysis
You are not audible,
of
sir.
the HSA.
Yeah,
Yeah,
it's okay now.
is I'm audible?
Yes, now you are audible.
Earlier I was not.
Yes, sir.
For few seconds you were not.
Okay, I'm sorry. So can I go ahead?
Yes, sir. Definitely.
Okay. So here we have can calculate from this the Stern-Wolmer equation constant, and the other equation that is needed is the by which we can calculate the KQ, and that KQ is intrinsic binding constant, and with this we can the value of KQ for the binding of compounds with HSA were of the order of 10 to the power 13, as we can see at different temperature when we see in this table. Okay, so KQ is coming in this direction. So therefore, it is evident that the quenching of HSA fluorescence was due to the formation of a complex with compound, and the mode of quenching is static or dynamic can be obtained by analyzing the dependency of KSV on temperature. If we go in the previous slide, we have taken the KSV values at different temperatures. Okay, the 308, 303, 298, and similarly we have done. So the pattern that obtained from the at various temperature shows whether the mode of a quenching is dynamic or static. Okay, so for static quenching, the KSV value decreases with increasing temperature. On the contrary, there is an increase in the values of KSV with a rise in temperature, as we can see in the chart in this table. This trend confirms that both the compounds mode of quenching with HSA fluorescence is static rather than dynamic. So after this, we had use the another equation, that is a modified Stern-Wolmer equation, in which we can correlate the Ka, the binding constant, and as well as the number of sign. It gives us what we are getting, like Ka is this binding constant, and this is the number of binding site, means how it is binding and what is the number of binding sites. So the modifiers of the interaction with HSA values of binding parameters are shows that the number of binding site for both ligands was near to 1 to all steady temperature, and the value of Ka was of the order of 10 to the power 4 to 10 to the power 5 per mole. And now for the complex compound 2, it shows that it is coming in the range of 10 to the power 5 to 10 to the power 6, with a moderately high binding affinity for these compounds. Further, we're using the help of Van 't Hoff equation, we also calculate the thermodynamics of these compounds, and these thermodynamics, the interaction of both the negative value of delta H denote the exothermic nature, and the positive value of delta H shows the endothermic nature of the, as it is well known. The interaction of both the compounds was endothermic in nature. We have also calculated the Gibbs free energy. The negative value of free energy change for both the compounds confirms the interaction was spontaneous. Yeah. Sorry. Now, the next experiment that we performed was FRET. FRET is Foster resonance energy transfer. It's a valuable tool to study the extent of energy transfer and distance between two compounds. The FRET theory states that when the absorption spectra of the receptor molecules overlap with the imaging spectra of the donor molecule. Both molecules are in proximity, that is less than 8 nanometer. So as we see that the overlap is shown in figures, and the parameter obtained from the FRET analysis shows that there is an the efficiency of the energy transfer from HSA to compounds were 37.3% and 61.2% respectively. Similarly, R for the interaction of compound were obtained as 2.84 and 2.49 respectively. Interestingly, a large value of R then R naught suggested that the interaction of both compound with HSA was static quenching with the formation of ground state complexes. This also goes with the, as we had done before, the other data. So the next experiment that we performed was 3D fluorescence. The 3D fluorescence imaging spectra is used to analyze the conformational and the structural alteration caused in protein due to the interaction with the molecule or the compound that we use. The 3D fluorescence imaging of free HSA and HSA plus compound are shown in the figure, and the intensity of the image signal also enlisted. So the two prominent peaks in the 3D fluorescence of a protein are attributed to Rayleigh scattering and aromatic amino acids. The peak intensity of native HSA at peak 1 and peak 2 were recorded as at 5949 arbitrary unit and 3870 arbitrary respectively. In the presence of compound, the quenching in fluorescence intensity at both the peaks was observed, which denotes the formation of complex and an overall decrease in HSA diameter. Moreover, the quenching of fluorescence at peak 2 indicates the conformation alteration around tyrosine and tryptophan of the proton. This 3D spectral analysis further validates the conformational alteration induced in HSA due to the interaction of the compound with HSA. These are the peaks and data that shows that there is an quenching that shows like with compound 2, it is coming to 81 to 65 like that. After that, we used to go for computational chemistry like molecular docking with some of the free softwares. In this, I will go a bit quickly, maybe I'm taking too long. So in this, what we take the molecules and take the protein, like in this case, we have the HSA, and we had docked it with the compound, and we found that the complex compound 1 and 2 preferentially binds to the Sudlow side or subdomain 2. There are many pockets that are available in the proteins, and that are specific for every compound. Similarly, the Sudlow side 2, subdomain 2, where they were formed several hydrophobic interactions with several amino acids like glutathione, GLU153, CYS200, CYS249, histidine 242, lysine 195, lysine 199, and arginine 410, valine 433, cysteine 437, alanine 449, leucine 453, and arginine 485, with the residue of the Sudlow side 1 and side 2 binding site cavity. Both compounds were seen like that. And similarly, we have shown some of the interactions that take non-covalent interaction that take with these compounds and the ligands that we had taken place. These are just an amino benzimidazole group with this, and we have shown all the interactions that are possible. By this, we stop our instrumental, or what you call the binding studies, and we go for in vitro assessment of these molecules for cytotoxicity. Just a second, my light is with sensor, so it has stopped, so I cannot see properly. Yeah. So with the MTT assay, we used to go with the standard drugs and compare them when the compound shows less than 10 microgram per ml activity, IC50 value, sorry, less than 10 microgram, then we consider them the active ones. Okay. So when we found any of them and we go for we got the compound that have the relatively good cytotoxicity with the IC50 less than 10 microgram, we go for small basic experiments with ROS generation with the help of dyes that you can see. We had done with the MCF7, and we have exposed our compound for 24 hours. And then we observed that there is a fluorescent light that shows that these compounds are showing ROS generation. Similarly, we go for glutathione level. This glutathione level in cancer cells is having very important role, as it is supposed to be have level depleted, the level increased of glutathione in the cancer cells. So we study our compounds by treating the cells and study the glutathione level. And what I think that there is a mistake with 5 milligram and 10. It is 5 microgram, 10 microgram, and 15 microgram. So we had studied the level of glutathione. The level of the glutathione goes down upon treatment in a concentration dependent pattern for fashion. Similarly, we have also studied the lipid profile, lipid peroxidation level for these with MCF7, the breast cancer cell lines. And with the exposure of our drug, we found that these the level of LPO increases. That is to be needed if there is any type of ROS generation. So one of the ROS that we are studying is glutathione. The other one is lipid peroxidation. After this, we go with our collaborators with the help of flow FACS analysis, like flow cytometry. We go with different type of kits, and there is a kit that gives us the detail of cell cycle arrest. It is said that the most anti-cancer metal-based therapeutic employs their activity as potential activity via DNA damage and cell cycle perturbation. In the phase of the cell cycle, the cell's relative population can be determined using tropium iodide, a fluorescent dye, to fix the cells and analyze the flow cytometry. Analyzed by flow cytometry. In this case, we have noticed that the G2M phase has twofold of DNA or DNA compared to G1 phase, and the actively replicating DNA is found in the S phase. Therefore, we investigated these molecule cytotoxicity against this one. I have taken with my paper in Dalton, and this is with the in the with A549 cancer cell line, and that treats shows this one as the control one, and then these three are after the treatment with 3 micromolar, 7 micromolar, and this one is 10 micromolar concentration of the compound. So in this at lower concentration, the G2 phase showed a significant increase in the cell to 34.71, leading to G2 arrest. However, the cisplatin after DNA binding is known to stall the cell in the S and G2M phase. Further, we studied the apoptotic assay. The apoptotic assay is just what you call how the cells are dying. They are dying by necrosis or dying by apoptosis. So there is an assay that is called as annexin VFTIC PI assay. In this, we treat with the kits by tropium iodide, and this process is called as dual staining on cancer cell, and it also been analyzed by the flow cytometry. In this case, what we notice is that at concentration of 7 micromolar, 31% and 20.8% are the early apoptotic and apoptotic cells. Then there is, you can see that these are the phases, like A1 is for necrosis, A2 is for apoptosis, A3, this one is for viable cells, and A4 is the early apoptotic cells. You can see that in the first one, that viable cells, dots shows the number of viable cells, means the live cells. Upon treatment, with only 3 micromolar concentration, there is a decrease in this. It comes around 90% usually in control. I think I forgot to put the control. So with 3 micromolar, there is a decrease in viable cells, mean the live cells. So they are coming towards the apoptotic and early apoptotic cells. Then what we are getting is, when we treated with 7 micromolar, the quantity of viable cell reduces, the quantity of early apoptotic and apoptotic cell increased. But the quantity of necrose cells also increases, but not up to that extent. Then when we increase the concentration to 10 micromolar, the concentration or the cells are more in the apoptotic region and early apoptotic region. If we count both of them, it will come around 80% of the cells at 10 micromolar got killed by apoptotic pattern, and very small section of 13% is coming like in necrosis. So after this, we have studied these cells for various other stains that will show us whether the cells are, the compounds is entering our cells or not. This staining basically was done just to see that there is the entry of our molecule or something is happening inside the cell, or just it is from the outside. So as we can see, the A is the control one of that is stained with HS 33342 A549 cells. After the treatment of one of our compound that has been published in Dalton in 2024, for treatment of 24-hour, A is the control one. Here we take the lesser concentration, that is so that the cells should not get killed completely. So we had taken the 3 micromolar and 7 micromolar. And in this, when we stain with it and treat it with our molecule, what we found is that there's a fragmentation of there's a nuclear fragmentation and margination of the nucleus, means the nucleus is coming toward the center, toward the periphery. It is coming toward the end of the cell walls, and this type of reflection means that the appearance of the cells are morely done by the apoptotic mode of cell death. Then we have done check the morphological changes by dual staining, that is done by acridine orange and ethidium bromide. These are more towards what you call as I am forgetting that. Let me see, what is it? Yeah. So in these type of staining, they are really done whether it decrease the cell viability after treatment is due to apoptosis. AO can acridine orange can permeate into both live and dead cells to stain all nucleated cells and excite green fluorescence, while PI only enters dead cells with damaged membranes and generate red fluorescence. This one is the red one, and these are the live cells, so they are giving green fluorescence. Okay, so in general, the cytological changes are of 4 types in the AOEB stain: uniformly green, fluorescent, highly organized, structured, that is viable. You can say most of them are viable here, means the live cell. The second one is the bright green patches and fragments, perinuclear chromatin condensation, and early apoptotic, like this. In this case, there are something like this. These are green cell, but there are coming of chromatins or fibers that you can see. Pictures are not that much clear, but you have to means just imagine that these are showing some small sections. Then there is an orange to red fluorescent nuclei that is chromatin condensed and fragmented late apoptotic, like these orange cells. Yeah, these orange cells are like apoptotic and late apoptotic cells. Orange to red, no chromatin fragmentation indication, swollen large size and necrotic cells. Like in this, there is nothing but the cells broken down itself without any staining. So in the percentage of viable cells are decreased in this, and it shows that the quantitative analysis shows that the analysis of apoptotic and necrosis cells revealed that a high percentage of apoptotic cells and necrotic cells will increase concentration. With increased concentration, there is an increase in apoptotic cells and necrotic cells. Then we had done an dye that is for vascular organelles, and that is with acridine orange. For vesicular organelles, this induction is called as autophagy induction. This is done for the autophagy associated cell death is the typical non-apoptotic cell death, which is characterized by cytoplasmic vacuoles. Okay, we investigated with our compound the cell death associated with that of with the autophagy pathway, again, was used as fluorescent marker for the acidic vesicular organelles. In this case, both the treated cells showed more AVOs in the cytoplasmic perinuclear region as an autophagy indicator. Then we had taken the membrane potential of these membrane, because if there is some drug that is entering into the cell, it should increase the potential of the membrane, and that is being stained with rhodamine 1, 2, 3 for this. So as we see with the fluorescent intensity, as it increases, as it decreases, it shows that the potential on the membrane has increased. After this, when our molecule clearly passes everything, then we go for gene expression. Like in this case, we study the proliferation of these cells with different concentration. As we have seen that the control was having a number of cells around 44 lakhs. Then we had treated with 3 micromolar of our compound, then 5 micromolar of compound, and these compound reduced to around 1 lakh. With this treatment, we have isolated the messenger RNA of these cells treated with our compound and studied the genes with the help of the KIT RT-PCR, and messenger RNA expression was studied. And we have particularly taken the genes that are MMP2 and TGF beta, mostly because that was the only available genes with our collaborator. So they have studied it, and they have found that with the concentration dependent pattern, both MMP2 and the TGF beta decreases the control gene expression that was. So it is relating with the gene also and helping with this. So that's really strange. I think my slide, I have missed my conclusion slide, but anyway, at present, I can, with this studies, we can see that there is a large scope for these ruthenium compounds to go for it, and needs to be explored as much as possible with different biological active compounds. And with the different techniques, it can make us easy to contact with the pharmaceutical industry and attract them by doing as much as possible laboratory or the tests that are possible with us as academicians. That is what we can do. And now I would like to thank all for inviting me. And yeah, some of the papers I wanted to show, but at present, I think I had a different presentation that I had. But anyway, I can check if there is something that I have left. This is the paper that I had published with Professor Aditya Kakkar and Deepti Gupta in 2013 in organometallics, and we had done good work in that. And that was with the ruthenium. It's a bimetallic compound with ruthenium marine and organotin. So that we had published before, but at present I want to see my file that I'm not getting in it. Just a few seconds, maybe this one. No. Anyway, thank you so much. I missed a few, sorry for interruption because I have missed a few slides that I don't have. I'm really thankful for the organizing committee of this conference, Deshbandhu College, for inviting me, Professor Rajiv Agrawal, the principal, the organizing committee, especially Dr. Deepti, Dr. Manisha Singer, the chairman. Thank you so much. Thank you everyone for listening to my work. Thank you.
Thank you, Dr. Khan, for your the wonderful talk. And I'm just happy to inform you, that I have already gone through your paper, that is Dalton transaction of 2021, and I specifically, I'm very much interested in vitro study what you have done. So it's a very nicely presented and well written paper. And yes, in future, definitely we will again collaborate with you and work because Dr. Umesh is also pursuing some of the work in this area. Yeah. So I'm very happy that, the design and new organic motif and their respective ruthenium compounds, what you have studied very nicely and very well designed work, and specifically methodology I like much. I'm not a chemistry person, but yes, being a biology side, I could say you have, most of the work what you have done and very nicely, and that in vitro analysis you done. So thank you so much and congratulate you for wonderful work. And
thank you so much for your
yeah,
I really appreciate for your kind words, and it's just a start, and we are just in the beginning of our research. And as you know that with the funding and everything is going down day by day, but we are just trying small sections that can help us. And just I try to sum up few of my papers just to make the new researchers an idea that we can, as an academician also, we can collaborate and work. As you can see with my presentation that I had a big collaboration with many of international people, like from Italy to Netherlands to Germany to US to Portugal, because other than that, we in the university, it is not easy to work in research, and we have to collaborate. And
basically, when you work in interdisciplinary area, definitely, sir, we need a big team and definitely different people from different specialized area. Then only we can come out with very good work which you have come out. So
yeah,
yes.
exactly. But also, as we all know that, as being, I have done my PhD from India also and from the university as well. So there is a big difficulty while limitations with different limited resources. So we should not be feeling any any type of hurdle by collaborating with anyone, because in our people usually thinks that collaboration is not that much good. But I think if you have to learn and to excel in your field, you have to go with the new dimensions with different collaborators. So
Research
thank you
journey
so much.
always we need to learn from each other. We need to collaborate. We need to, then only you can ideation, and then finally we'll have better outcome. Yeah. Thank you so much,
Sure.
Dr. Khan. And definitely we'll welcome you physically also when you are in India. So we can make a plan and you should visit our small, the college. As other I could say small, but yes, one of the oldest college of Delhi University, which
As I told you that previously I had worked with Dr. Deepti and Dr. Rita Kakkar in my PhD days long back. But that was a nice experience, and thank you for your invitation.
Yeah.
Surely I will come and meet you people when I will be in India. Thank you.
Thank you, Dr. Khan. Thank you. Over to you, Dr. Parul.
Thank you, sir.
Any questions? Do we have time to take question if anyone is interested? Or any question in the chat box? But I think
I think there is one question in the chat box.
Okay, sorry, I cannot see that.
Yeah, I can read if you wish.
Yeah, please go ahead.
There is a question from Rahul K. I see new drugs are being discovered for treating cancer on a regular basis, and most of these drugs have multiple side effects.
Yeah.
So what should we be done so that the drug directly reaches the target cancer cells without affecting other cells? And how to reduce the side effect, and further how to enhance the bioavailability without increasing the drug dosage?
Yeah, that is what I have used a term several times, that you have to think of a biocompatible molecule, okay, and a molecule that should carry your molecule to the specific site. The problem is that molecule that you have synthesized is going to the target that you want to, and that can be done with the help of any carrier. Like in this case, in our molecules, we have taken the help of protein HSA, okay, that is also a carrier to the molecule. But in this case, we have to be, this is the most important point that the person has asked. I could not see his name, but anyway, Rahul K. Yes, Rahul has asked really a good question, but still the people are searching the answers for it. But the only thing that we can do is with the literature and with as much as you read, you will go step by step ahead, and you will came to know that how can we reduce than previous drugs. Like cisplatin was having too much toxicity. Now the drugs that are in market, they are less toxic. So with the time, advancements comes, and that is where we publish, and that is recognized with the time. So you will learn when you work with it, and with the time you will know how you can reduce it, because there are many issues, like it is coming with the selectivity also, and also with the compatibility of the molecule. Because if you have the compatible molecule and it is moving towards the target, whether it is reaching to the target or not, still it is a matter of concern, and many groups are working over it. There are fields like drug carriers and many things that are introduced and in progress. Yes, the question is good, I should say, and it will be hopefully in the future it will answer us, but still we are trying. So thanks, Rahul, for asking this question, and don't lose hope. We can still do better.
Thank you, sir.
Thank you.
And kindly accept a token of appreciation from Deshbandhu College.
Dr. Rais Ahmed Khan, sir, please stop your sharing.
Okay. I could not do it, maybe.
At the top. Sir, at the top.
Yeah.
Red color box.
Is it done?
No.
No. Oh, yeah, I think. Now it's okay?
Yeah.
Yes, sir. Thank you, sir.
Thank you so much.
Dr. Ashwini, it's not visible.
Ma'am, I think it's a network issue. Just wait.
Yeah, it's visible. Kindly accept it, sir.
Yeah, thank you so much for this memento and the certificate. I really appreciate it. And
you,
I'm
sir.
again thankful to all of you, all the organizing team, the chairman, the dean, and etc. Whoever is involved in this process, and especially the organizing team, because they have to go with a lot of difficulties while arranging such an event. Thank you so much,
Thank you, sir.
Parul.
Thank
for
you.
the-
Thank you,
Thank
sir.
you, Dr.
Bye-bye.
Khan.
Thank you.
Thank you, sir. Thanks so much.
So it's the time to conclude the session. First of all, kindly accept my apologies for technical glitches. I extend my sincere thanks to both the speakers, Professor Niranjan Parajuli and Professor Rais Heman, for enlightening us. I am grateful to our chairpersons, Dr. Manisha Sengar and Dr. Indrakant K. Singh, for sparing their times. My sincere thanks to all the participants for making the session successful. And now I request Dr. Ashwini to present a token of appreciation to our chairpersons.
Thank you.
Thank you, Chairman, sir.
Thank you. Thank you so much for this
Thank you, ma'am.
opportunity.
Thank you, sir.
Thank you, Dr. Parul, Dr.
Thank you,
Umesh,
sir.
and complete team for organizing this event.
Thank you very much, sir. And there is an announcement for the participants. Due to the time constraint and due to the back-to-back session without any break, we don't have interactive session right now. And the group photograph would be after the validatory function. Please keep on join on the digital platform. We will reassemble again at 2 p.m. sharp after lunch. Thank you very much
ये
once
वीडियो
again.
ऑफ कर दो पार्टिसिपेंट्स के। ये सुरभि पांडे अपना करे ही जा रही है। वीडियो ऑडियो इनके ना म्यूट कर देना। देखो
Thank
ना
you.
वो अपना
Thank you,
सजती है और फिर खड़ी हो जाती है। ठीक है, क्योंकि रावत सर बोल देंगे, पहले ही करके रखो इसको ना। सेटिंग मोनिका से पूछ लो, लीना को भी पता है। जैसे ही एंटर होंगे सबको म्यूट और सबके वीडियो ऑफ। ठीक है, चलो अब लंच के बाद मिलते हैं। रिलैक्स।
Yes, we will reassemble at 2
एक
p.m.
बार चैट
sharp.
बॉक्स में डाल दो तुम भी। मैंने भी डाल दिया फीडबैक फॉर्म, एक बार तुम भी डाल दो। हाँ, ओके।
Thank you.
Feedback form is posted in the chat box. It's mandatory to fill the feedback form.
And it is for all 4 lectures, pre-lunch session. All 4
It's for
lectures are
pre-lunch
covered
session.
in that. Yes. And for next session, we request all participants to please keep yourself muted and put your videos off, because the next speaker is our Dean Examinations, Professor D.S. Rawat. And, it is disturbing at times to the speaker. And feedback form for all 4 sessions is there. Kindly fill it to get the EAT certificate. So we rejoin at 2 p.m. again for the next session.